Are we ready to go? No, that's not good.
Yes, I can see. Yeah.
Okay. Good afternoon, everybody. Well, COBREG 2021 virtual has just started. Last year, COVID did us in. We couldn't meet. But this year, thanks to advancements which came as a result of COVID, we can now have virtual meetings, which we believe will now be part of it, where even if we are gathered together, people can still give their presentations either live or recorded by video. So going forward next year, we hope to have a much more interactive COBREG conference. Well, this year we sent out a call and the response was good. So here we are today to listen to various talks from various areas. We have a total of 18 talks. 2 of them are keynotes and the rest are the short presentations. So I can see quite a number of people have logged on and most of the speakers are ready. And most of them have also tested their slides. So we are not hoping to have any hitches once we start. So all things being equal, I'd like to welcome you all to COBREG 21 virtual. And for the next 2 days, let's share ideas, let's share links, let's collaborate to move science forward, especially for our up and coming scientists in Ghana. They need to know that there is hope for science in Ghana. So I always like the idea of COBREG to give them the necessary hope and mentorship. So I hope members will take advantage of it to form links with other members and then establish collaboration. So welcome everybody, and I hope we all have a nice time. Thank you, George.
Thank you very much, Dr. Ayamfo, our Scientific Committee Chair. You've done, you and your committee have done a great job putting together this conference. So we look forward to these 2 days, today and tomorrow. We hope that we'll build on our tradition, our fine tradition of collaboration, capacity building, networking, and mentoring. Since 2007, this organization has come to be a force on our landscape. It all started with a discussion around the lunch table. And our keynote speaker here, Professor Jay, was present when we started discussing this. So many others have taken up the call. I mean, it was an idea whose time had come, and we have grown since. The state of science in Ghana has advanced some. We still have a long way to go, but we've made some strides towards the right direction. These last 2 years, we've been forced to limit our activities to online activities only. We had planned a series of workshops, but we've had to put those on hold for now. Our activities have mainly been webinars. Since COVID-19 has been the concern of the day, our more recent discussions have been centered around COVID-19. I'll just touch on the last 2. On the 1st of April, it was the 8th of April, actually. There was a webinar presentation by Drs. Joseph Boni and Karen Duker. looked at post-vaccination, what the fact that there are vaccines now available, what impact it has had on the course of the pandemic. And then on June 24th, we had our own secretary, Dr. Kodura Samuel Kusi, present to us on, and Dr. Arthur Kennedy here in the US, present to us on COVID-19 updates and perspectives. And so these were all very insightful sessions, and we learned a lot. But beyond these discussions of COVID-19, I just returned from Ghana. You know, life is going on. A lot of people are almost oblivious to the fact that we are in a pandemic. But the fact of the matter is that we have come a long way since the beginning of 2020, when we didn't know what to expect. We have vaccines. We are able to conduct our lives to a large extent as normally as can be. Models have changed. I am working remotely sometimes and going to my lab and office at other times. Students have had to adapt to new models of learning, and time will tell whether these innovations will have the same effect as the more traditional ones. So, ladies and gentlemen, without much ado, we look forward to these next 2 days. I will hand over to the presenter of the keynote address, Professor James Ajay. And I'd like to pause here. Akwesi, do I get to introduce Professor Ajay, or there's a moderator who takes over? Dr. Yamfo?
Yeah, George. You hand over to Dr. Ekuya Kakari. She will introduce, she's the moderator and will introduce the.
All right, thank you very much. So Dr. Ekuya Kakari, the ball is in your court now. Thank you so much.
All right, thank you very much. So good afternoon to everyone. I'm Ekuya Kakari, and I'm delighted to be introducing our first keynote speaker for this conference in the person of Professor James Ajay. He is the chair of stem cell research and regenerative medicine at the Heinrich Heine University. Actually, this is German, so please forgive me if I got the pronunciation wrong. He's involved in systems biology-based projects, both at the national and international level, using iPSCs. He has won several awards, and his stem cells from urine project won the SIGA Rheinischer Innovations Prize for award-winning business idea in 2018. Okay, so the entire biography of Prof is part of the program outline, and I don't want to bore you going through all of that. I want to share my personal experience with Prof. We met at a DAAD conference in 2016, where we had different scientific lectures running concurrently. And out of curiosity, I just decided to join the stem cell lecture where he was going to give a talk. And it was one of the most informative and comprehensive lectures I have ever attended. In fact, the moderator had to force the session to a close because we had gone past questioning and answer time into coffee break, and the session was getting even more interesting. And actually, we were the only group that came out late for coffee break. Today, Prof will be speaking on pluripotent stem cells from bench to bedside. And I can guarantee you that it's going to be a very enlightening session. And so with great pleasure, I present to you a mentor, an outstanding researcher, and an inspiration to young scientists like myself, Professor James Ajay. You're welcome, sir.
Thank you very much. First of all, I'd like to Of course, thank you, Dr. Kahari, for the introduction. We met a long time ago. Here I am again. Also like to also mention, reiterate what Professor Akwa Mensah mentioned. We met a long time ago. I think this was in Vienna, if I'm right, having lunch. And here we are again. It's been quite a while. I'd like to also thank the organizer, Dr. Anyinfo, for the invitation. I think I first spoke at this meeting, the Ghana Biomed Convention, I think it was 2013 in Legon.
Yeah.
This was when Dr. Drucker, yeah, exactly, a long time ago. So the prodigal son is back. So this time around, let's have some humor. So as the title suggests, pluripotent stem cells from bench to bedside. So what I'm going to do is basically just give you a flavor of the activities we're involved in. I'm not going to go into too much mechanistic details. A lot of the work has been published, some not, but just to let you see what we do using pluripotent stem cells. So I'll describe what they are and the applications. So first of all, a disclaimer. So these are biotech startups which are co-founded, still looking for investors if any of you are interested. So basically, my talk or my research embraces embryogenesis right through to aging. So embryogenesis meaning the union of egg and sperm to derive a one-cell embryo, two-cell, all the way to the blastocyst, hatching, then placenta, pregnancy and birth, etc. So thereon is disease. So the research I was doing in the late 90s focused on these areas here, working on gene expression during early human development. So there we worked together with IVF clinics, and I was in London at that time, at King's College Hospital and Royal Free. Well, of course, you can do this type of research in human, but you'd stop at the blastocyst stage. So how do you go beyond to understand human development or disease? So to do that, of course, you need to use pluripotent stem cells. So I'll describe what these are, how you derive them, and how they can be used for disease modeling. So the first pluripotent stem cell to be derived was in 1998 by James Thomson. Okay, these are embryonic stem cells. Like I mentioned, they are derived from the human blastocyst, which is this structure here, and in particular, this cell type here called the inner cell mass. So in normal development, so the inner cell mass is what forms the fetus, and the surrounding trophectoderm forms the placenta and the extra embryonic structures. But of course, one can isolate these inner cell mass cells and culture them indefinitely if you use the right conditions, and you culture them as embryonic stem cells, I can see here as colonies. And these were, of course, in the days where cultures were on mouse embryonic fibroblast cells. These are the feeder cells, and they secreted factors that maintain these cells. Okay, so embryonic stem cells in culture, these cells actually do not exist in the human body. These are, I like to call them an in vitro artifact, because such a cell does not exist in our body. So the embryonic stem cells, by nature, by virtue, can differentiate into cell types representative of the three germ layers. So ectoderm, mesoderm, endoderm. So ectoderm, of course, brain, skin, adrenal. We talk about mesoderm, heart, kidney, blood, bone, endoderm, liver. And in my institute, we research mainly on brain, kidney, bone, liver, using pluripotent stem cells. Of course, if you work with pluripotent stem cells, which I mentioned, this comes from a human blastocyst. This you can only have access to if you have an IVF, an in vitro fertilization program running, and if the parents or the couple give approval for you to use surplus embryos for research. Of course, this is frowned upon in many countries, especially countries where they're fully Catholic. The other countries are much more lenient. In Germany, this is actually quite old, nearly 10, 12 years now. But this is just the landscape at that time, let's say 12 years ago, of embryonic stem cell research within Germany. And before anyone works with embryonic stem cells in Germany, which is still the case, you need to file an application which is approved by the National Ethical Board, right? And if they deem your project useful, you get approval. So this is a long time ago. I was living in Berlin at that time. I'm now based here in North Rhine-Westphalia, in Düsseldorf. And Düsseldorf is actually a hotbed for this type of research in Germany. So you can see there are 19 approved projects at that time. The number has really gone up. So if you work with embryonic stem cells, you're also going against the church, at least the Catholic Church. Okay, so this is an article that came out a while back. I was in my 40s, young, full of hair. And basically, it was 5 years after embryonic stem cells were allowed to come into Germany. So we had to get a renewal to allow us to work with these cells. So this became a big debate. And of course, I stood out like a sore thumb. And I was in the media, etc., talking about what we need to do, what we cannot do. And of course, opposition was the church. But at least they oppose, but they cannot stop this type of research going on. So the problem aforementioned, embryonic stem cells. So in the UK, you can, in France, you can, in Poland, for instance, very religious Catholic, you cannot. So in the US, of course, you can. So the way to bypass this limitation, which is linked to ethics, is what we call induced pluripotent stem cells. So what are these cells? So the first reports of embryonic stem cells came from Shinya Yamanaka's lab, and also soon after by James Thomson. So James Thomson is the one who derived the first human embryonic stem cell line. And you can see, basically, you take any, in the beginning, they always use adult skin cells. So you take a sponge of the skin, culture out the fibroblasts, and then you introduce these 4 transcription factors, OCT4, SOX2, KLF4, c-MYC. In the case of James Thomson, it was OCT4, SOX2, non-organ Lin28. But of course, this cocktail is more potent. Why? Because it's more oncogenic. You have OCT and SOX in both, but you have c-MYC, which makes this cocktail combination more oncogenic, or let's say much better at deriving iPS cells. So this is just showing the 2012 Nobel Prize for Medicine awarded to Yamanaka, and of course shared with John Gordon, who years before had shown that the xenopus, the African toad, the egg of the African toad, the nuclear content can use to reprogram somatic cells. But what Yamanaka did, he defined just 4 factors. John Gordon said the entire egg. Right, so how do you do this process? How do you reprogram an adult cell? This is quite an old slide when you use viruses. So you'd prepare the viral particles expressing these transcription factors. So everything I'll talk about is about the Yamanaka factors, okay? So viral particles are made, you add that to your fibroblast cells, you transduce them, you wait till you get colonies, and you pick them further. And this is just showing a time schedule. So here, let's say day 0, we add the viral particles encoding the 4 factors, and these are dermal fibroblasts, actually human fetal foreskin fibroblasts, which you can buy. Then after you add the factors, you'd see between 3 and 7 days, tight clusters. So here you see what we call mesenchymal epithelial transition. So fibroblasts are mesenchymal cells, iPS cells are E-cadherin positive epithelial cells, sorry. So you can see here the tight clustering gets much tighter, and then you get a colony. You can see it better on this slide. And here, these are some early publications, 2012, I think 2013, where at that time, I was on a trip to figure out which cell types can be used for reprogramming. And for me, then the cells had to be very young. And also, you can imagine if you use sun-exposed skin cells, the tendency is that when you age, the cells are aging, there are chromosomal aberrations, they are more sensitive to ROS, etc. So young cells are better. So this was the aim at that time. So we managed to collaborate with the maternity ward, this when I was in Berlin, where we obtained amniotic fluid, which is the fluid that bathes the fetus, and also chorionic villi cells, which along the lining of the placenta. And with these, of course, when you keep them in culture, they proliferate very fast, but you hit Hayflick's limit. Every primary cell undergoes replicative senescence. They just stop growing, they fall apart. But if you add the 4 factors, you make an iPS cells, unlimited self-renewal. And in these 2 papers, we could turn these iPS cells, which are made from amniotic fluid cells and chorionic villi, we can turn them into placenta-like cells. We brought them back in development. So actually to get us to understand how a placenta is made, for instance. So once you make the iPS, you have to characterize these. So by overexpressing the 4 transcription factors, you activate then the endogenous genes, OCT4, SOX2, NANOG, and other cell surface proteins. Okay, and then these then keep the cells under this pluripotent state. And if they are pluripotent, they should be able to, like I showed in the early slide, they should be able, under spontaneous differentiation, form cell types representative of 3 germ layers. So you have the endoderms, alpha fetoprotein, SOX17 are cells you see, or positive cells you see in the liver. Then you see smooth muscle actin, you see in the heart, right, brachyuria as well. Then of course, ectoderm, you're talking about brain cells, so beta-3 tubulin PAC6. Of course, pluripotent stem cells, if transplanted into an immune compromised mouse, after about 2 to 3 months, you get teratomas. And this is the pathologist analysis of this histology. You can see within the teratoma, you have neural tissue, cartilage, bone, etc., which again tells you these are pluripotent. And that is the reason why taking these cells into therapy, you need to make sure that you have differentiated the donor cell free from any cell which is pluripotent. If you don't, you'd get this, okay? But we know, and you'd see induced pluripotent stem cells and embryonic stem cells are already in the clinic.
Take my drug.
Right. So this is a paper we published which summarizes the senescence-related mitochondrial ROS pathway is repressed in iPS cells. Another test you do is called the pluri test. So here we take these UM51 cells, which are adult cells. We reprogram them into iPS. Then we compare the transcriptome of these cells to embryonic stem cells, H1, H9, and you see they cluster together. They're more closer together. H1 and H9, 0.92% identical, transcriptome H9, and that again there. So with all these tests in place, you can confidently say you have an embryonic stem cell. Okay, so again, most of this being published a while back. We've also published other papers, mechanistic as well. So you could just go into PubMed, look it up. If you can't get hold of the papers, you can email me and I can send you the PDFs. So my quest for sources of cells which are easy to obtain. Now, if you take a skin biopsy, this can be painful. We had a project with aged individuals, which was a problem to get skin biopsies because of inability to heal. So my quest was, how do I find cells which can be used for reprogramming? And a colleague of mine in China showed that you can reprogram and make iPS cells using cells derived from urine. And this is what we reproduced. So here, I'll give you a guess whose urine that is. So basically, when you take urine, you spin them down. And we devised a medium which can be used to select these cells, okay? And this is the basis of one of the spin-offs we've made. They're not, although it sounds very simplistic, it's not easy to do. It's all based on the medium. So you just need urine. You've identified this cell population. These cell populations have the characteristics of mesenchymal stem cells, meaning they can be differentiated into adipocytes, which is fat, chondrocytes, and also osteoblasts, so bone and cartilage. And when we compare the secretome of this UM51 and UM27, which are urine-derived MSCs, if we compare the secretome of these to fetal MSCs, you see they all secrete similar factors, which tells you that these cells have regenerative potential. Look at the transcriptome of these cells and look at the gene ontologies, it tells you these are all kidney. So what we have are indeed kidney cells, right? And that's just showing that these cells can also be reprogrammed very efficiently, very fast. They meet the criteria for pluripotency, and the UM51 is me. I was 51 at that time. So in the lab, most of us have our iPS cells in culture. So most of the talks you'd see, some of the slides, you'd see that most of them actually use my iPS cells, right? And I think in the future, we hope that this will be the way forward, that we'll have iPS from every individual and that you can use for therapy or drug screening. So they just summarize this. You have urine, you can get these cells. These cells are kidney cells by virtue of the expression of 6,2-WT1 cited. They also form tubular cells. Okay, you can make iPS out of them. You can make kidney organoids, you can make brain organoids. You can turn these iPS cells into liver to express albumin and also into brain expressing MAP2. And these are some of the papers related to our urine work. This is in revision. We showed that we can use this cellular model, not the iPS, but the urine cells to study the renin-angiotensin system. So the pathways that leads to blood pressure, for instance. So to summarize, iPS cells, you need somatic cells. It undergoes epigenetic reprogramming to have the iPS, which can be differentiated into different cell types, which in the future can be used for autologous transfer into a patient. But also you can do, the heat transfer, not autologous, but matching, donor matching, and as you do with blood, okay, the HLA matching and transplantation. And also for in vitro research, so disease research, drug screening, genetics, developmental studies. So now I'm going to talk about disease research. So how we use these iPS cells to study disease. Okay, so the first I'll talk about is Alzheimer's disease. I'm not going to go into details because of the time factor, just to say that we have the familial Alzheimer's disease and we have the sporadic. So the familial is gene-driven mutation, okay, and that's about 5% of those suffering from Alzheimer's. And the sporadic form is about 95%. And there is environmental risk factors and aging, yeah, with the ApoE genetic background. And of course, the APP cascade, if you follow that, you have APP produced, but this is processed into the A-beta oligomers, okay, A-beta 40, 42, and this forms the amyloid plaque, which subsequently or time leads to cognitive dysfunction. So we try to understand all these processes using iPS cells. And in particular, what I'll talk about is one of these. So if you do genome-wide association studies, you can identify various genes which you can associate with this disease. So then you have to confirm it. So we did TREM2, and there we derived iPS cells, etc. I'll talk about that. But just here showing that there are several papers where iPS cells have been used to model Alzheimer's disease, okay? Using it for drug screening, etc. So I'll talk about the TREM2. So the TREM2, which is trigger receptor expressed on myeloid cells, in particular microglia, right? This function of this has been shown to be important in multiple neurodegenerative diseases, AD, Parkinson's, okay? And what we work on is the mutant. So individuals that have the TREM2 arginine 47 histidine variant. So we want to understand what this mutation does in terms of the disease. How does it impart the disease? Of course, TREM2 expressed on microglia cells, and microglia we know are needed for phagocytosis, debris clearance, okay, in neuroimmune, right? So here's just a schematic showing it's published, okay? So we have your iPS cells, we undergo a series of developmental steps, last 80 days in culture. Then you get this elaborate neuronal network, which consists of GFAP positive cells, so astrocytes, and also neurons. We never get oligodendrocytes or microglia in this protocol, in this 2D protocol. So this can also be a limitation because you don't have the full complement of brain cells, but at least one can work with these. These cells also have action potentials using patch clamping and also the MIR chips, right? So these are the MIR chip, multi-electrode arrays. And of course, you can stimulate the cells and you can measure activity. So for instance, now, when we compare the Alzheimer's disease TREM2 variant, the transcriptome of these iPS cells compared to healthy controls, right? And just to summarize, we look at gene ontology, so molecular function, biological processes, cellular component, But if you look at the pathways which we identify, several, I just made this quite short, but you see pathways related to drug metabolism, of course, some reactive ligand receptor interactions. But what actually recently caught my eye through a collaboration with Dr. Kakari is malaria, African trypanosomias, and also complement, so immune-related features. Okay, and of course, if you look, because we're talking about the TREM2 variant, which is immune-related, we see a lot of immune-related processes, biological processes, differentially expressed or altered in the disease state. So this just to summarize this work. So we had these iPS cells, we had age match control, we derived these neuronal networks, we stimulated the cells with A beta and analyzed APP. We also identified metabolic dysregulation, impaired phagocytosis, altered inflammatory response. Okay, again, there are numerous publications on that from my lab. Okay, I'm not going to talk about NDS that much, just to say that this is a rare disease, Nijmegen breakage syndrome, which is localized eastern part of Europe, so Poland in particular. In Germany, it's difficult to get these individuals suffering from this because most people are affected by NBS. So it's a mutation in Nibrin, which is involved in a DNA damage response pathway. So individuals affected do not live beyond their teens. They die very early. So we've been working on this. One of PhD students, 2017, made iPS cells out of it. We used it to screen for antioxidants. We later published together with a group in Israel, so Nissim Benevistri and Michael Goldberg, to show that chromosome instability and molecular defects in iPS from these patients. Okay, and we summarize it here. So basically, patients that have this mutation have deregulated p53. And of course, if you have deregulated p53, you have cancer, you have all sorts. But these patients also have microcephaly, so small heads. So this is work which is, we've submitted the revision with Soraya Martins, just finished a PhD early this year. So of course, again, we showed impaired p53. And what was interesting, she made brain organoids out of these. But what we noticed is that if you look here, so this would be the brain organoid of two controls and these NBS 8 and 1. The NBS 8 is a heterozygous mutation, NBS 1 is homozygous mutation. What we see, or we'd like to think we see, so what the reviewers are saying, is that with the NBS 8, the homozygous mutation, the organoids tend to be smaller. They always tend to be smaller in diameter, which would reflect the microcephaly phenotype. We've done a lot of molecular analysis. I'm not going to go through that. So now I come to the kidney. So you've seen this slide already. You've seen these papers. So what Lisa has been doing, so part of her PhD work, got another year to go, she's derived kidney organoids. It's quite funny to see because these are actually my kidneys in the dish. We use this to study kidney injury. So one of the substances we've been using, amongst others, is PAN, so puromycin aminonucleoside, which is a nephrotoxin also used to induce nephropathy in rodents. So she makes these organoids, and when we do the H&E stain of sections, you can see typical structures you'd see in a kidney. So you see the glomerular structures here. You also see tubules. Okay, and when we stain LTL, it's a tubular marker. Alpha-actinin 4 is a glomerular marker. And you can see overlap, but you can see a region where you see more of the glomerular positive cells. And when we add PAN, what we see is really a collapse of the cytoskeleton, amongst others, right? So when you add PAN, this is gamma H2X staining of the cells, you see an increased expression of this DNA damage marker, gamma H2X. You see on the cellular level, you see it also in the Western blot. Likewise, we look at p53, it's upregulated because there's damage to the DNA, and also caspase 3 is also increased, and that's the quantification. And the pathways which are activated, of course, by addition of PAN or inducing nephro damage is p53, as you'd expect. You'd see so genotoxicity pathways coming up, or biological processes, should I say. You also see indications for cell junction reorganization, so the cytoskeleton being disrupted. And of course, downregulated our cell cycle, DNA packaging, etc. We also studied liver disease, okay, non-alcoholic fatty liver disease. It's a lifestyle disease mainly. Bad diet, lack of exercise, the Homer Simpson style of life. You have your normal liver, you have a fatty liver full of fat, leads to cirrhotic liver and cancer. It's bad nutrition, sugar, sweet, sedentary lifestyle, lot of fat. Certain drugs can also induce this. This is typically you see from a healthy individual, the section through the liver, and the section of the liver of someone with fatty liver disease. You can see these white vacuoles, these are actually lipid droplets, where fat droplets, okay? So it's just a bad lifestyle. And we could induce this by just making the cells and adding oleic acid, adding fat to the cells. And this is what Nino has done. And we tried to understand more mechanistically what's going on, okay? So basically, again, the process follows typical developmental cues. So you have your iPS cells, you go to definitive endoderm, hepatic endoderm, and then to the liver, hepatocyte-like cells. And being liver cells, they express alpha-fetoprotein, they express albumin, E-cadherin, HNF4 alpha, etc., various other markers. Also, as liver cells, they secrete urea, they synthesize and secrete urea, and you can see that here, and also have cytochrome P450 activity. This is just to show a bit complex, but just to show it's a complex disease. It's a complex metabolic disease. But here, when we have the hepatocyte-like cells, we add oleic acid, you see the fat droplets formed, okay? And these are CLIN2, which surround the fat droplets. So normally healthy individuals have high levels of adiponectin. And adiponectin, of course, is a hub. It's involved in several metabolic pathways, AMPK, gluconeogenesis, yeah, PPR alpha, gamma, insulin as well. Yeah, so diseased individuals have very low level of adiponectin. So our idea was, if you add this small molecule adiporon, can you then substitute for the lack of presence of adiponectin? And this is what the paper was about. Right, so like I said, I haven't gone into detail because of time, but just to give you a flavor what you can do with iPS cells. So in terms of regenerative medicine, we published this paper in 2019, won so many awards, because what we did here is we took human foreskin fibroblast-derived iPS cells, we made MSCs out of these, and then used this to cure or to treat pigs with a injury. So, and what we did here is these are these iPS cells where we differentiate to MSCs. So basically, using this approach, you don't have to go to the bone marrow to get MSCs. You can use an iPS cell, which comes from urine, comes from skin, and turn these into MSCs. And of course, you can see alizarin red staining, and then also the typical MSC-related, bone-related markers are up. So this was a very big study, nearly took 2 years. So we had 9 these Göttinger mini pigs, they weigh between 100 and 120 kilos. We worked together with the surgery department, with the surgeons, with anesthetists, et cetera, full preclinical study. So this is the injury they produce. And then they have the CPG, is calcium phosphate granules. So you bathe the cells in CPG. Then we had bathe the cells, CPG with the IMSCs, CPGs with bone marrow concentrate, which comes from the pig, okay? And then the autograft, which is MSCs from the pigs, like autologous. So just believe me, I believe them. So the yellow arrows points to newly formed bone. So this is when you use the granules alone. There is some, not much. When we use the IMSCs, we see also some newly formed bone. And then when you do the autologous, so the bone marrow concentrate with the CPGs, well, of course, these are the MSCs coming from the pig, you see full, okay? Also CT scans, right? And basically what this tells us is that the autograft is the best. The bone marrow concentrates, these are all autologous, but the iPS approach actually was better than just having the concentrated, the calcium phosphate granules alone. So our next step now is to use these urine-derived MSCs, which can make bone cells, and repeat these experiments with the view that this would be an approach for the future. This just summarizes the approach that what could be happening is that these MSCs, when put into the site of injury, they probably differentiate into bone cells, or there's paracrine signaling, they release factors which aids resident MSCs to recover and do what they're supposed to do. We also use the same approach to cure rats suffering from jaundice, okay? So this also works. Now, we cannot go without talking about COVID-19. So basically, iPS cells have also been used to study COVID-19 infection and downstream. There are several papers, no one there. What we did beginning of the year or last year, we downloaded data sets where they infected human lung epithelial cells with the SARS-CoV-2 virus. They did a transcriptome, NGS. We took that, reanalyzed it, and came up with the schematic that the virus needs the receptor ACE2, an accessory protein, the TMP-RSS. There's several of these. Enters the cells, replicates, then of course damage occurs when they release. You have a cytokine storm, you have ROS production, ultimately leading to cell death. Another paper which is still under review, it's been a very difficult one, very challenging one, but here we used, and actually I must compliment Ghana for this, because here we had several initiatives in different countries. When you have your viral isolates, they sequence this and they deposit the sequence in repository. So in Ghana, of course, Noguchi doing this work. So you have access, you can have access to all the sequence information. That's what we did, we clustered them. And what we could follow through the mutations is that, of course, the infection started from China, we all know that, Wuhan, okay? And then it migrated into the rest of the world. So we know here that the original mutation went to the UK, from the UK into Ghana, right, and Nigeria, from France to Senegal. So it was very clear that, again, saying that within the UK, Ghana, Nigeria being former British colonies, you have more people from the British-speaking colonies living in the UK than, yeah. But anyway, regardless, the point to make is that data sets are available. We can take them, we can reanalyze them and do our own experiments. So here I've highlighted Ghana. So this was in our revision. They told us, well, but since then, there's also the Delta strain or the Delta variant. So we have to reanalyze that. And you can see indeed that in Ghana, the sequencing of the Delta strain was around 2021 in April. Okay, so all this information we have, hopefully we get this paper accepted soon. And what we're doing now is we have the spike protein, which we are overexpressing in kidney cells. Of course, kidney cells also express the ACE2 receptor, and then to look downstream. So in terms of therapy, there are several trials going on. Some are recruiting, most of these are recruiting for cardiomyopathy. This is in France, for ALS, heart failure, several diseases, both IPS as well. So really, these cells have really got to the clinic and it's looking very good. And in Japan already 2 years ago, they managed to cure a lady with macular degeneration using her iPS cells, which they differentiated to retinal epithelial pigmented cells. So iPS cells, ES cells are in the clinic. And I hope that one day we will be able to do this type of research in Africa, let's say in Ghana. It's not just about the research, it's not about going for therapy, but these are also cells that can be used for screening for drugs. Yeah, because big pharma in the past were using cell lines which are transformed, but now if you look at the African population, we have various diseases which we can look at, we can derive such models for drug screening and also for research. And I'd just like to end with this slide again. And what I want you to remember is urine is precious. We think about it as waste material, but really it is not. And this is what we are championing and really pushing forward. So we have the protocol which works beautifully, reproducible. The only problem is that the urine has to be fresh. 24 hours maximum, but fresh better. And that is the only drawback. So we're finding ways, for instance, if I want to have urine sent from Ghana, the time it arrives, I mean, things you have to keep room temperature, because if it gets cold, the urea crystallizes out and this affects. So these are the drawbacks with this approach. It has to be fresh and not the medium. Also, is a bit of a trademark, but we're hoping that I can transfer this research to other parts of the world, well, to Ghana. When I was other parts, I mean Ghana, right? So I'd like to end here. by acknowledging members of the lab, past and present, the various funding bodies over the years, collaborators in Ghana. You know who this is. So Patrick Kobiner, I've been working with in Legon, but there he has these the extracts from fungus, where we've already shown that these have anti-cancer effects, okay? And through that work, we applied to the DAAD, and we have a student, Benedine Tua, who's going to start in January. Should have started this year because of COVID. It's been transferred to next year. She's going to start her PhD program with us, working with Patrick. Also, Osmond was a master student in my lab and now works in Heidelberg doing his PhD. And these are the various lab members. So I'd like to take this opportunity to say thank you once again to the organizers. And I promise I'm going to take part in this meeting every year from now on. And hopefully when COVID is okay next year, I'll be in Ghana for that. So thank you very much.
Well, thank you very much, Prof. We wish we could clap. So yeah, that was a very great talk. So now we're going to invite some questions. So if anyone has a question to ask Prof, you could type it in the chat session, or you could raise your hands, and then we'll be glad to take your questions.
I'm curious, if it's okay. Are there any insights that you would share, Prof, regarding the factors in the urine that could be responsible? I'm just wondering if other body fluids could perform as well as urine.
In which process?
And if that, yes.
So in which process?
Oh, in inducing
pluripotency. Okay, to get cells. Yes. So though, you know, about 10 years ago, there's 1 or 2 papers came out saying that the hair follicles you can do, yeah. So there, although you can pluck your hair, if you get the intact follicle, you can expand cells from that, but it's so inefficient, so that did not work. But you can also use blood. So you can use lymphocytes from blood. But then again, if you take blood, then you have to process the blood. Then you have to get these lymphocytes, which you need several cytokines, growth factors to scale up, right? So that's possible. So most people still tend to use skin because fibroblasts are very robust. Urine, I would say, is the easiest. It sounds easy, but not every, I mean, there are companies that sell medium, which if you use on this urine, what you isolate are the differentiated tubular cells, which do not proliferate. So it's not that efficient for reprogramming. The medium which we use in our method, you get the progenitor cells defined by 6,2-WT1 and cited. And these are much easier to reprogram. So in principle, any part of your body where you can get cells, at least 100 to 200,000 cells, you can reprogram them.
Thank you.
All right, so I see Dr. Nyamfon's hand up. So, Doc, you can unmute
thank
yourself.
you. Looking at where we are now, does this mean we are getting to the end of embryonic stem cells? Because that was the one that was causing trouble. Now that we can get it from adults and reprogram into a lot of cells.
Right.
And 2, how close are we to an actual cure using the regenerative medicine? I mean, I can see animals. How close are we to an actual human cure using any of your methods?
Oh, all right. Okay, so I'll tackle the first. I mean, I'd like to say that embryonic stems or IPS, is getting closer to the point where you can say that IPS is a substitute for embryonic stem cells. Okay, but end of the day, embryonic stem cells are always the best because these have not been genetically modified. But we've got to an era now where IPS cells are much better. Why? Because, of course, it's much easier to derive IPS cells. There are no ethical implications. It's unlimited. To the embryonic stem cells, you need to have through IVF. And also with the IPS approach, it's much easier to study disease because if you have an individual with a gene-driven disease, of course, you can get the person's skin cells or the person's urine. Okay. For instance, we've collaborated with a clinic in Aachen where they have patients suffering from a liver disease. They have this mutation in alpha-1 antitrypsin. So we just got urine from these patients. We've made iPS cells, and now we're studying the effect of the mutation on these patients. So the iPS, I would say, is the way forward. It is the way forward. It's in the clinic, like I showed you in the slides. Okay. And in terms of therapy, it's happening. It's happened. People have been cured. Like I said to you, in Japan, for macular degeneration, in Japan, the Japanese are really in the forefront because they believe in the system, and their ethics concerning this type of work is not as strong as the rest of the world. So what they're planning for next year, using iPS cells and using them making liver buds or liver organoids, making brain organoids, they intend to use this to cure Parkinson's and other liver diseases. So really, there is a lot of success stories. Of course, bone marrow-derived MSCs have been used for a very long time. This goes on, but the iPS approach is really moving forward. In Paris, they've shown for myocardial infarction, at least the first 2 weeks, that they use embryonic stem cells. They're also using iPS cells. Also in Boston for type 1 diabetes, there's also several approvals now using iPS cells for Parkinson's, etc. So really, we're at the point where these cells actually can be used for healing. Of course, it's something which at the moment is for the elite, right? It's not routine, but hopefully it might get to that point where it might end up being routine. But I think for me, the iPS approach best is for using as a screening platform to screen for drugs, right? You know, that is the major impact you can have with these cells. For instance, we had a company approached us, a big pharma approached us for Uricell, thinking that we had thousands and thousands of these kidney stem cells, which they could use for their screening approach, right, for kidney-related diseases. So unfortunately, we don't have that many because we're still building up. But Jessica, my dream really would be to come to Ghana, set up this urine-derived stem cells for a whole host of individuals, both for research, for the intellectual property as well, and as a screening platform for diseases. And there are big pharma, Novartis, etc., who are waiting to have such, not just in Africa, but also in Europe. And that is why we set up Uricell, which we're gradually trying to, so these type of cells have really big potential, not just for regenerative medicine, but also for drug screening, for toxicology studies.
Okay, so probably there's a question in the chat session. This is coming from Dr. Stephen Amwa. He says, nice presentation. Which is an easier approach with your technique, forming new kidneys or deriving podocytes, and that the fixed glomerular filtration barrier for renal diseases?
Okay, I'm looking at the question now. So,
okay,
which is an easier approach, forming new kidneys or deriving podocytes? Okay, so first of all, I'm trying to understand what you mean. So for instance, you can still see my screen, right?
Yes.
Yes, okay. So the podocytes, you can easily get podocytes from these urine-derived renal progenitor cells. It's a very simple differentiation protocol. Within, let's say, 3 to 4 days, you have a dish full of podocytes, okay? Now, when you say forming new kidneys, I don't know what you mean by forming new kidneys, but deriving podocytes is very simple, very straightforward. And that's the problem, because so far, podocytes have been very rare and difficult to get. Most people doing podocytes or even having these kidney cells, epithelial cells, they take a kidney biopsy and then isolate these cells. Totally inefficient. It's like with the hepatocytes, you take a liver biopsy, etc. So using the iPS approach, especially for the kidney, using this approach is really the way forward. The next question is, and hope that the fix the glomerular filtration barrier for renal diseases. These are all things we're thinking of doing. So if you want to communicate with me, if you're an expert in all this on the glomerular filtration barrier, rate, etc., I'll be really happy to hear from you. But this is the direction we're getting now. So using these, and actually most of my research now, I'm moving into the kidney, because there is an avenue which hasn't been touched. And so there we really can follow, like I said to you, we have the podocytes, we have we can follow the whole pathway that leads to blood pressure. So we add angiotensin 2, we're looking at the angiotensin receptor 1 or 2, blocking them. There are ACE inhibitors. So basically the medication you use for treating, for lowering blood pressure, we run all these on these cells to understand really how this medication works. So this is the platform we have, and it's the direction I'm going. So for sure, Stephen, if you have excellent ideas you want to share with me, work together on the kidney, the glomerular filtration barrier and rate, by all means, please do.
Okay, thank you very much, Prof. So I don't see any further questions. Oh, there is a hand up, sorry. So Dr. Nyamful again. Doc? Dr. Nyamful? Okay,
Oh yeah.
let's see.
Sorry, I was muted.
Okay.
Oh, you forgot to put his hand down.
If there's another question, I'll let him come first, since I've already taken a 10.
All right, so there is one from the attendees. I saw a hand up, but it's no longer there. So Doc, you can go ahead. Dr. Nicholas Thomford. Yeah, so we'll start with Dr. Thomford first.
Yeah, Prof, interesting presentation.
Thank you.
My interest is, I joined a little late. I saw one of the slides where I think you had introduced some mutations, heterozygous and homozygous mutations, where I think it affected the sizes of the organoids that were formed. Yes. So I was only wondering, in terms of, and I think you acknowledged it, that in terms of African populations, we have quite a diverse genetic makeup. Yeah. So when we want to probably introduce something like this in terms of using iPSCs to model diseases and probably eventually bring out some therapeutic effects, how practical would that one be? Most especially looking at the fact that our jurisdiction, we do not have probably the setup for these things.
Yeah. So basically, okay. So first of all, these NBS patients have microcephaly, very small brains, right? And this is what we could see. But of course, because this is a very rare disease and very difficult getting enough patients, one of the problems we've had from reviewers is that you only have 2 individuals. We did not introduce the mutations. These are the mutations already, germline mutations. So we just got the skin cells from these patients, from these children. Right, yeah, we didn't introduce it. But now using genome editing, CRISPR, you can introduce your own mutations. So for instance, you can take, let's say, control 1. You can introduce the mutation in there. So then you'd have even a much better control because you'd have the same genetic background as a wild type and the same genetic background as a mutation. So that is what people are doing now using CRISPR technology. Okay, so the other question is how to bring this to Africa, other parts of the world. I mean, it's very costly. This is really, it's not like culturing Heck or HeLa. I mean, this is very, very expensive research. To maintain these cells with the growth factors, let's face it, even some labs in Europe can't afford to do this type of research. So it really boils down to money, infrastructure, know-how you can learn. Yeah, know-how one can learn, but really it's not a big problem. It's just boils down to money and infrastructure for the IPS related work. But for the kidney related work, the process and the culturing of these cells, it's not that expensive compared to the iPS cells. And that is something which can be done if you have the infrastructure for cell culture. So a good cell culture laboratory, a good setup is possible. So like I said, I'm on a mission. So next year, hopefully, come and visit some labs and see what we can do.
All right, so Dr. Nyamful, we have 2 questions in the chat session, but then because your hand is up, Dr. Nyamful, you can ask your question.
I think his mic is off.
Yeah, it's still okay. So then there is another question from Dr. Amwa. He says, in the use of iPSCs for neurological disorders, how is the progress on these neurons integrating into the neuronal circuit and improving behavior such as in Alzheimer's disease?
Okay, in the use of iPS for neural disorders, how is the progress on these neurons integrated into the neural circuit improving? Oh yeah, so there are several animal models. So there are several rodent models which have shown, which have turned out with very good, interesting results, that the human iPS-derived neurons really integrate into the brain of these mice and actually improves cognitive features, etc. So there's no doubt whatsoever that this does not work. It really does work in animal models. So the question now is transferring this to human. And like I said, there is now approval with Bayer, the Bayer, the pharmaceutical company in Germany, together with a biotech company based in New York, to really use iPS cells to treat Parkinson patients. Okay, so really it has got to the point where we see improvement. It's happening, right? We are really there. Okay, and the next is another question after that.
Yeah. That is coming from Dr. Kwame Bonsafo, I think. He says, great presentation, Prof. Could you expand on the applications in human reproduction and pregnancy? I'm a gynecologist.
Great. So actually, I stopped doing that work. So, the early papers I talked to you about where we made the iPS cells from chorionic villi and amniotic fluid. There, for instance, we could show that these iPS cells, we can turn them into placenta cells. So if you can do that, you can study a lot of placenta-related diseases and conditions, right? In terms of human reproduction, now this is very controversial. I mean, at least in mice, they've made iPS from the tail tip. So they took the tail tip of a mouse, isolated fibroblasts, made iPS cells, turned these iPS into eggs and into sperm, right? And really created a mouse, right? Or they've taken the sperm, taken the eggs from another mouse, and using ICSI, inserting the sperm. So in the mouse, they've recreated another mouse. Human, they keep talking, they're doing this. It should not be done. I think that is completely wrong. It should not do that. Yeah, but in terms of studying gynecology related or pregnancy related or screening for pregnancy related drugs, etc., the iPS is a great platform for that, because you can easily make, like I've shown you, you can easily make placenta cells. For instance, we made these placenta cells, we could actually measure HCG. I mean, it really works. You can make placenta cells out of it. Yeah, I mean, the applications are numerous. It's just a matter of finding the right protocol and the right questions.
Okay.
Right.
So there is one question again
from Mohammed.
Dr. Mohammed, yeah.
Mahdi.
He
Okay.
says, thank you, Prof, for such a presentation. Can you use iPS cells to induce mutation in beta islet cells?
Right. So maybe your question is a bit confusing. So first of all, you can derive beta islet cells using iPS. That's been done. You can do it. But what you say to induce mutation in these cells, okay, if my understanding is correct, what you're trying to say is that, which I hope it is, that if we take a healthy iPS cell line, you can use genome editing to induce a mutation and then derive the beta islet cells out of that, right? But the iPS itself process does not induce the mutation. So let's get that right. The process of deriving an iPS does not induce mutation. What it does induce is chromosomal aberrations. So you always screen for colonies which do not have aberrations. But the iPS cells do not induce mutations. Okay, so that's a correction. But you can use iPS cells from a healthy individual. And if you know of mutations, or you know the typical transcription factor which drive beta islet cell formation, you can delete that, mutate that, and see if indeed when mutated, you cannot have islet cells formed. All right. So iPS cells do not induce mutations in these cells, but rather you can use iPS cells, use genome editing to create a mutation and study the cell type downstream.
Okay. So no further questions. So yes, as always, an enlightening and informative talk, Prof, we are very grateful for your time and for sharing your research with us. I know that there will be a lot of collaborations stemming from this talk. And so we will end the session for the keynote address. And I think Dr. Tomford will take over from me.
Okay, thank you very much for your attention. Thank you. So,
okay, Dr. Tomford.
Here, I am here. Welcome once again to GBC Conference 2021. Our next session is made of short talks of about 15 minutes. And basically this side of the talks would focus on genomics and clinical research. Each presenter will have 15 minutes. Then after the whole presentation, we would have after the presentation, we would have question time. For the first presenter, we have Ekuya Kakari from the Department of Biomedical Sciences, University of Cape Coast in Ghana. And she would be presenting on blood-based transcriptome analysis of P. falciparum patients. So without much ado, Ekuya Kakari, we hand over the baton to you for the next 15 minutes.
Thank you very much. So I'm going to be sharing my screen now. Please, is my screen visible?
It is.
Yes,
yes,
we can see.
Yes, it is. We can see.
Okay. All right. So thank you very much, Dr. Comfort, for the introduction. I'm Ekuya Kakari from the Department of Biomedical Sciences. And this afternoon, my talk is going to focus on genes and cellular pathways, which are upregulated during Plasmodium falciparum malaria using blood transcriptome analysis from previous data. Now, I might say that this work was done in collaboration with the Institute for Stem Cell Research and Regenerative Medicine, Universitäts Klinikum Düsseldorf. So malaria remains one of the most deadly diseases in the world, accounting for over 300,000 deaths in 2019. And of the 5 Plasmodium species that infect humans, Plasmodium falciparum remains the most virulent, causing the most severe forms of malaria. So falciparum invasion inside the host with the subsequent release of parasitic toxins, notably the hemozoin, triggers a series of host responses. So based on the symptoms of a patient and parasitological examinations using thick and thin blood smears, falciparum malaria can mainly be grouped into uncomplicated, or what others term mild malaria, non-cerebral severe malaria, which also includes severe anemia malaria, and cerebral malaria. Now, very few studies have used blood transcriptome analysis to determine gene expression profile in these 3 falciparum malaria. So the main aim of our research was to compare the changes that occur at the molecular level between the 3 falciparum malaria based on previous transcriptome data. So to do this, what we did was to pull transcriptome data from previous publications from Thiam et al. And Bolt et al. Then we had to pre-process this data to get rid of every form of batch and background effect. And then we went on to determine the pathways and genes which are upregulated using the Kyoto Encyclopedia for Genes and Genomes and Gene Ontology database. Now, I'll be presenting some findings from our manuscript, which is under revision, currently under revision. So turning our attention to our results. First, we constructed a diagram. We wanted to know the genes which are mutually expressed between mild malaria, severe malaria, and cerebral malaria, and those ones which were exclusively expressed. Now, of the genes which were exclusively expressed, we found that CM patients, that's cerebral malaria patients, had the least number of genes as compared to mild malaria and severe malaria. Now, of the genes which were mutually expressed in the 3-falciparum malaria, majority were involved with inflammatory response, cellular, and ER stress. Now, what we found interesting was the upregulation of genes associated with Alzheimer's disease, namely the amyloid precursor protein and O2 gene, which is involved in mental retardation. Now, for CM, this doesn't come as a surprise because it has implications on the brain. But for these genes in mild malaria and severe malaria suggest that there is some impairment going on in these malaria states as well. So next, we zoom in on the genes which are exclusively expressed in the 3-falciparum malaria. So we turn our attention now to mild malaria. So for the pathways, we measured the pathways which are upregulated in mild malaria, and we discovered those for lung cancer, base excision repair, DNA repair, prion disease, and Parkinson's disease. Now, these two came as a surprise because it is contradictory to previous findings where neurodegenerative pathways were associated with cerebral malaria and not mild malaria. So the question is, what was going on? And we really had to dig the literature to look for any information regarding mild malaria and cognitive or brain impairment. And well, I was glad when I came across some of these publications which had reported cognitive impairment in some mild malaria patients. The only exception is that in mild malaria patients, the impairment is transient, okay, as opposed to what is seen in cerebral malaria patients. So the question was, why is cognitive impairment in mild malaria transient? So we went on to determine the genes which are upregulated in mild malaria, and we found those which regulate DNA damage response, blood vessel remodeling, IL-10, and mismatch repair. So it looks like in mild malaria, there is a counteractive response by the host to combat the deleterious effect of falciparum malaria. And this may account for the transient cognitive impairment in mild malaria patients. So turning our attention now to severe malaria. Now, the genes which were upregulated in this malaria condition were those involved in cellular response to DNA damage, negative regulation of cytoskeletal organization, MAP3 kinase activation, amyloid beta clearance, and protein kinase activity. Now, when these genes are upregulated in a cell, it points to severe cellular injury. It points to severe cellular injury, and there are also hallmarks for certain neurodegenerative diseases. And so it wasn't surprising when we came across this publication by Hickson et al., where they reported long-term behavioral problems in children with severe malaria who also had acute kidney injury. So next, we move on to cerebral malaria. And there we found genes associated with signal transduction in the absence of a ligand, which suggests an aberrant signal transduction, neutrophil degranulation, which is associated with blood-brain barrier and neuronal damage. So these two are suggestive of a mechanistic clue to neuronal damage in cerebral malaria. What was interesting was the upregulation of genes which repress gene expression. So we had negative regulation of gene expression, proximal promoter DNA binding transcription repressor. And this could explain why we found CM patients having the least number of gene expression in our Venn diagram. And so the question was, which are the genes that are being upregulated and which are those which are being downregulated in cerebral malaria? And so for the genes which were upregulated in cerebral malaria, majority of them were associated with inflammatory response, cellular injury and death. And then when we turn attention to those which were downregulated in CM, we found homeostatic process, neuroprotection, and metabolism. They were all downregulated in cerebral malaria patients. And so it looks like in CM, the balance just swings in favor of damaging host responses as opposed to the protective ones. And that's why the cognitive impairment or brain impairment in CM patients is long-term. And so to conclude my talk for this afternoon, we have shown from this study that p-falciparum-induced inflammation and cellular stress mechanisms may impair brain function in mild, severe, and cerebral malaria. And the neurological deficits that are predominantly reported in CM cases could be attributed to the downregulation of various genes involved in cellular function. And p-falciparum malaria and some neurodegenerative diseases share similar biological processes. And so the results of this study actually correlated with a publication from Professor Ajayi's lab, which we had a discussion on. So they used an iPSC model to simulate late onset Alzheimer's disease. And then they had to construct an Alzheimer's gene regulatory network. Now, guess who showed up as one of the significant pathways in the network? Our good friend malaria showed up again. And so zooming in on the genes which were upregulated in malaria, we found a central cluster located around genes associated with Alzheimer's disease, that's buttressing our earlier observation that AD or neurodegenerative diseases and malaria share very similar biological processes. And so what we want to do next is to model cerebral malaria using iPSCs. So from Prof's lab, we are going to get iPSCs with the R47, TREM2 mutation and control cells. We want to grow them to 3D neurospheres where they show some pan and mature neuronal markers like MAP2, SMI, and MIN. And then we are going to be using hemozoin. So this is the main toxin that is involved in falciparum pathology. And so we use this toxin and simulate cerebral malaria in iPSCs. And then we're going to be looking for some AD enzymes and genes like gamma secretase. So this enzyme is very necessary to produce the pathogenic amyloid beta. So in the presence of hemozoin, what is happening to gamma secretase activity? Then we'll look at inflammatory responses and how they modulate all of these gene expression. ApoE, so this protein is involved in lipid metabolism. And a recent finding from one group in Seattle discovered that using statin, I think that lowers cholesterol levels is very protective against the detrimental effect of cerebral malaria. So the question is, what is happening to ApoE during cerebral malaria pathogenesis? And so with this, I would like to say thank you to Professor James Ajayi for initiating this collaboration and providing resources for the study, and also to Dr. Rook. Actually, he was the one that did all the bioinformatic stuff because I'm not a bioinformatician at all, I'm a neuroscientist. So, and thank you all for your attention.
All right, thank you very much, Ekuya, for the wonderful presentation. We would ask questions later, so if you have any questions, kindly take note of them so that at the end of the presentations for this session, we will call for that. Next on our list, we have is it Franklin, who is from Warwick, yes, University of Ghana. And he is going to be talking about an investigation into cross-species potential of an AMA1-based malaria vaccine. So Franklin, for the next 15 minutes, we listen to you.
All right. Good afternoon, everyone. I hope you can see my screen. Hello?
Yes, we can see your screen. We don't see your face
Yes.
clearly, but we can see your screen. That's good. Thanks.
Okay, yeah. I'm Franklin Youngdem Nokem, and I'm going to give preliminary results from my research work titled An Investigation into the Cross-Species Potential of an AMA1-based malaria vaccine. And by way of introduction, malaria remains a disease of major public health concern. The WHO malaria report for 2020 estimated 229 million people infected with malaria in the year 2019. And by way of relating with this figure, that's 100 times the population of the people in Accra. And from these people, 409,000 died. It is significant for us to note that most of these mortality and morbidity cases are related to pregnant women and children. And of the total mortality, 67% of all those who died were children under 5 years, and about 822,000 were born low weight. This is aside the other major complications of malaria with regards to pregnancy and children under 5. And malaria is caused by species of the Plasmodium genus. Human malaria is caused by 5 parasites. The true human malaria causative organisms are Plasmodium falciparum, Plasmodium vivax, and these two have received so much attention in regards to intervention and research. The Plasmodium malariae and Plasmodium ovale have been neglected because they are known to cause very non-severe disease. And all these parasites are vectored by the female Anopheles mosquito. The fight against malaria intensified from the year 2000, and in 2019, the WHO assessed results from the various interventions which were introduced. And it is significant to note that with Plasmodium falciparum malaria, we've had a rapid decline from 2000. And from the year 2014, we've seen a store up to 2019. And this signals the need for additional interventions. But this is mainly for Plasmodium falciparum malaria and vivax. The challenges with the fight against malaria include diagnosis. And with diagnosis, mainly they are directed against Plasmodium falciparum, especially in this part of the world. And most of them are detecting HRP, the histidine-rich protein 2 or 3. And Dr. Amwa, who is listening, is one of the authority persons with HRP deletant parasites in Ghana. We have some of these parasites deleting the HRP protein, and therefore they cannot be detected in diagnosis. And also, with regards to vector and parasite resistance, they've been on the rise. And even with artemisinin, Southeast Asia is reporting a resistance to the main frontline active drug we are using to treat malaria now. With these, an effective malaria vaccine, as reasoned by numerous scientists, will fast-track mortality and morbidity reduction, especially among children below the age of 5. But also, we believe that malaria eradication should target non-falciparum species also. This is mainly because of their increasing prevalence, even as mono-infections. A study in Tanzania, that's in 2019, showed that although falciparum malaria is reducing, Plasmodium malaria and Plasmodium infections are increasing in prevalence. And although they were known to be part of mixed infections, now they are more in forms of mono-infections. And also, they are being recognized as causing severe disease now. Plasmodium malaria infections have been associated with kidney injury, and ovale is also known to lead to disease relapse. These parasites also, the non-falciparum malaria parasites also have low parasitemia. They are submicroscopic, and therefore they're able to produce gametocytes which sustain transmission. So if you target only non-falciparum, you have transmission with malaria and ovale, which you will miss out. And these parasites have different biology that's relevant to diagnosis, treatment, and prevalence. So it is in the regard that we should target these non-falciparum species that the study of the apical membrane antigen 1 It's a ringleader in malaria, in Plasmodium RBC invasion, and it is present across all Plasmodium species and even in apicomplexan species such as Toxoplasma. And it is critical in red blood cell invasion. Red blood cell invasion, the parasite needs to get into the red blood cell, otherwise it wouldn't survive. And it has been shown that antibodies against Plasmodium falciparum AM1 is capable of inhibiting parasite growth. With these attributes, the apical membrane antigen 1 has been developed into a leading malaria vaccine candidate. And one of the major hurdles is that it is polymorphic. Prominent solutions to these polymorphisms are epitope dilution phenomenon pioneered by Dr. Kwabia Samoa-Kusi and the diversity covering AM1 synthetic proteins. And usually such attempts focus on conserved regions of AM1, or they incorporate a lot of the polymorphisms which are present in nature. With AM1 being present in all Plasmodium species, is it possible to develop a malaria vaccine which can target at least 3 or which can offer cross-species protection? And we believe that this is possible with functional conservation, significant sequence and structural conservation of the AM1 proteins. And this led to the investigation of the structural and functional AM1 similarities and differences between Plasmodium falciparum, Plasmodium malariae, and ovale species. So to do this is on the backdrop that similar protein sequences will lead to similar protein structures and therefore similar protein functions. The first thing we sought to investigate is to check whether these 3 AM1 sequences are similar. And we align the sequences pulled from PlasmoDB using cluster omega. And when we did that, we realized that against Plasmodium falciparum, Plasmodium ovale has 51.3% similarity and malaria has 51.2% similarity. These are significant sequence similarities. And we also went further to check whether these sequence similarities translate into similar structures. This was done by pulling the X-ray crystallized crystallography solved structures of Plasmodium falciparum AM1 and Plasmodium vivax AM1 from PDB. And we predicted the structures of Plasmodium malariae AM1 and Plasmodium ovale AM1 using Swiss modeling. And these Plasmodium malariae AM1 and Plasmodium ovale AM1 structures have not been solved. And this further shows the extent of neglect of these 2 species because AM1 is a very advanced malaria vaccine candidate. And even just looking at this, you can clearly see that these 2 structures are very similar, but you can't just tell with the eye. So we further went to superimpose these structures with PF. We further went to superimpose these structures and we measure similarity using the TM score. The TM score is a range between 0 and 1, and it tells how similar 2 protein structures are. And as we can see with all the superimpositions, you have TM scores above 0.9. The TM score above 0.7 is very significant, which means that the 2 structures are very similar. So with a TM score of 0.9, these structures we are seeing are very, very similar. With these structures and their similarities, we had the confidence to express these proteins and study them and use them for functional analysis. So we express them using the baculovirus-mediated insect cell expression system. As we can see by Western blots, the protein was secreted and also in the pellet. Purification was done from the supernatant. And as we can see by SDSPH, the recombinant proteins were expressed and the purification process was very clean. And in-frame expression was confirmed using mass spec, as you can see for PM and PO. But unfortunately, we couldn't express PF AM1 fully, and we are still on that. So we had to borrow some from a lab to do the preliminary functional analysis. As you can see over here, this is PF AM1 reducing and non-reducing in an SDSPH gel. This is PM-AM1 running under reducing conditions and non-reducing in POAM1. The reducing conditions are run in the sense that the cysteine bonds are broken. So the proteins are linearized, and the non-reducing gel proteins are run still with their conformation, their native conformations intact. This is on the backdrop that some antibodies bind to conformational epitopes. So we want to see which antibodies bind to linear and which ones bind to conformational epitopes, to if there are some in a subsequent work. So we use a Western blot which featured an anti-His antibody to show how our proteins will be seen in Western blot, because the PM and the PO proteins were His-tagged. And this also confirms that our negative control, that's BSA, also worked. So with similar sequence structures, with similar protein structures, and on the backdrop that antibodies perform significant role in natural malaria immunity, we sought to determine the differential antibody binding to these three AM1 proteins. And the first of them was using purified IgG from immune individuals. And this purified IgG, we believe, because AM1 is immunogenic, will have antibodies directed against AM1. So as we can see for the first one, we have significant binding of antibodies to PFAM1 and PMAM1. But for PO, the band intensity is much lower. And these binding, we believe, may be due to cross-reactivity or due to specific infections by these parasites in humans. And we are yet to test that in further experiments. So monoclonals bind to specific epitopes, and with a lot of studies done with PFAM1, there are a lot of monoclonals which bind to PFAM1. So we sought to determine the extent of cross-reactivity using these monoclonals. And the first of them was N31D7, a monoclonal which was PFAM1 generated. And you can see it binds strongly to PFAM1. And it also binds to PMAM1, but for POAM1, it doesn't bind, as similar to what we saw in the purified IgG. So the next monoclonal we used was the 4G2 monoclonal. It was PFAM1 generated, and it binds to PFAM1, but it doesn't bind to any of the other AM1 proteins. And further work will be done in this regard using about 25 monoclonals to determine the extent of cross-reactivity of these proteins. So the next study was, the next aspect of this study was to determine seroprevalence, the proportions of antibodies in naturally acquired immunity. So we have people living in malaria endemic areas who are immune to severe malaria, who are immune to severe complications of the disease. And this has been proven to be partly as a result of acquired antibodies. So because AM1 is immunogenic and Ghana is predominantly Plasmodium falciparum transmission zone, we sought to determine the proportions which also recognize PMAM1 and POAM1. So the first was at Eduja using a sample size of 58, and these were mainly adults. And surprisingly, we realized that PMAM1 and POAM1 had higher antibody levels compared to PFAM1. This is not expected. And at another site, Aquachrome, we used both children and adult samples. And although there were differences, their medians were quite similar. We repeated the seroprevalence studies at Udumburum using a sample size of 280. And the same trend was observed with PMAM1 and POAM1 having higher antibody levels compared to PFAM1. So we thought that this was driven by a specific age group because malaria transmission and malaria complications are associated with age. So we broke the Budumburum sample sets down into 3 different age groups. And the first age group with 1 to 7 years, the same trend was observed. 8 to 14 years, the same trend was observed. And greater than 14 years, the same trend was also observed with Plasmodium malariae AM1 and Ovale AM1 having higher antibody levels compared to Falciparum AM1. The results from the seroprevalence studies will be investigated to confirm what we are seeing, because this is quite not expected, but this is what we have now. So further work will be to probe the AM1 proteins with a panel of anti-PFAM1 antibodies with map epitopes to determine the extent of cross-reactivity. And we'll do more of the seroprevalence studies, especially at higher transmission zones like in Tampo. Then we'll also estimate parasite prevalence, that's for the 3 Plasmodium parasites. And this can be correlated with the seroprevalence data we have, because if we have higher levels of PMAM1 and PO, higher levels of P malaria and P ovale transmission, then it will explain what we are seeing. Then we also do functional antibody inhibition assays, and it will complete this work. I'm grateful to my supervisors, Dr. Yawaniwe, Dr. Kojo Asamoah-Kusi, and I'm grateful to Professor Wandari for all the support, to my lab members, to TIBA, and to the molecular genetic cell biology thematic group at WA Group. And I'm grateful to WA Group for the fellowship. Thank you.
Thank you very much, Franklin, for the presentation and the work you're doing. Next, again, if you have any questions, kindly take notes, and at the end of the session, we'll ask you to ask your questions. Next is Stephen Amwa from the School of Medical Sciences, University of Cape Coast, and he'll be presenting on microRNA contributions to perinatal brain injury. So Stephen Amwa, the next 15 minutes is yours. You can unmute yourself and hopefully also put your video on.
Okay, so I'm actually using Dr. Nyamfo's computer simply because mine is having some problems. I hope you can see. Are you able to see my screen?
Yes, yes, yeah. See that.
Okay, great. Thank you then. Yeah, so this work is on microRNA contributions to perinatal brain injury, which is secondary to chorioamnionitis. This was done in Johns Hopkins School of Medicine, where I spent some time doing postdoc there. So perinatal brain injury is pretty much from the name. These are a cluster of conditions which are associated with brain injury during the time of birth. It could be very early, and it's very common in children who are born prematurely. So the preterm birth is associated to this. And also during this phenomenon, it's been observed that there is inflammation of the placenta. So if, for example, you could look at this, the image to the top right, you'd observe a developing fetus, and that is surrounded by the amniotic membrane. And then there is also the placenta that should be the site of, that should provide nourishment for this. You could either have maternal inflammation, in which case you'd have inflammatory agents crossing the placental membrane to then infect, to either infect or if affect the development trajectory of the development fetus, or you could even have pathogens tracking through the birth canal, which could also affect the fetus. And in all this, usually the amniotic membrane, mainly the amnion and then chorionic membranes, could then be inflamed. And when this is ongoing, various immune cells such as neutrophil and others also get accumulated by tracking in a chemotactic gradient. Inflammation is also able to elicit various molecules. And I'm interested in microRNAs, and some of these are called the inflammation-related microRNAs or inflammases. And these may either promote or seek to attenuate the impact of inflammation. And a number of microRNAs have been observed in these. These include the microRNA MIR223, which I'll be talking about a lot, MIR222, MIR458, have all been observed to be altered, mainly upregulated during chorioamnionitis, which I'll be calling chorio as an abbreviation. And the study of microRNAs in the context of chorioamnionitis may shed some more light on the post-transcriptional mechanisms at work. So what are microRNAs? MicroRNAs are a cluster of genes which constitute what previously was called junk genomes. These microRNA, these genes do not code for proteins. However, they've been observed to have various important functions, which I'll very soon showed, as a lot of literature is indicating. For the biogenesis, they could either be derived independently from intergenic regions of the DNA or spliced from intronic regions of protein coding genes. And either pathway, there is a precursor called the pre-microRNA, which is a hairpin structured precursor. This is exported from the nucleus into the cytoplasm, where there are further processes to cleave the duplex structure into, to cleave the hairpin structure into a duplex structure, which is then unwound. And then one of them, called the mature microRNA, approximately about 22 nucleotide, is incorporated into the RNA-induced silencing complex. So the mature microRNA serves as a template, and then it's used to scan for various mRNAs from the 3' UTR seeking for complementarity. And when there is a success, that mRNA is sequestered from being translated and subjected to decay. So this shows the important post-transcriptional role of mature microRNA. And it's been estimated that about 2/3 of mRNAs are regulated by various microRNAs. So these are important genes which have been observed to affect almost any cell in humans and other species. So to our methods. So for the method, we use timed pregnant Sprague Dally rats shipped from Charles Rivers, and then at gestational day E18, they were injected with 4 microliters of LPS to induce the chorioamnionitis. And then a day later, some of these were harvested, so E19, and this harvest was on the brain and also the placenta, as well as E29. The rats were born at P0, and that is usually E22 or E23. And for the rats, their 3rd trimester, part of it is outside the uterus, so that is usually up to P7. And then some were allowed to grow up to P21, which is equivalent to the human young toddler age. And for the chorioamnionitis induction, an abdominal incision is made, and then the fetuses are exposed, and then LPS is then injected into the, this is occluded for 60 minutes to induce ischemia, hypoxia ischemia, after which the LPS, which is in Evans blue, is then administered. And after that, the fetuses are returned to the abdominal cavity, and then this is sutured, and these are closely monitored to be able to observe any outcome. After this, RNA is isolated from both the placenta and also the brain, and then cDNA synthesis is performed for the microRNA and also the mRNAs, and then qPCR is used to amplify these genes. So to the results. As earlier indicated, the microRNA MIR223 has been observed to be highly expressed. So we were interested in the placenta, and we observed that one day following the chorioamnionitis induction, we observed elevation in the MIR223 within the choreo group as compared to the sham-treated rats. And then 3 days later, this upregulation was still evident. So we're now interested in, okay, we've observed upregulation in the microRNA MIR223. What happens within the inflammatory genes? And don't worry, I will just run through this. So we observed that in CXCR2, which is predominantly expressed in neutrophils as well as other immune genes, this was also upregulated in the choreo-treated rats. And then when we look at its ligand, CXCL1, this was also significantly upregulated. We looked at CCR2, which is predominantly expressed in monocytes and macrophages. This was also upregulated. NLPR3, which is part of the inflammasome, was also significantly upregulated at the mRNA level. We look at TNF-alpha, also was upregulated in the choreo group. And when we look at NF-kappa beta, that was also upregulated. And finally, IL-6 was upregulated in the choreo group. These are all pro-inflammatory genes. So we were interested also in the anti-inflammatory, in which case IL-10 we observed was significantly upregulated, and it is trying to compensate for the very highly inflamed environment of the placenta. So when we got this information, we're wondering, would this inflammation still continue in the E21? And we observed that it still continues in E21, that is 3 days after the induction of chorioamnionitis, except for CXCR2, which did not change. So having had this exciting finding, we were wondering, would there be any correlation between the microRNA miR223 and then these inflammatory genes? And when we look at that, you can clearly see that there is a significant positive correlation between the microRNA miR223 in the placenta at E19 with CXCR2, CXCL1, NLPR3, TNF alpha, NF kappa beta, IL-6, and then IL-10, except for the CCR2. And when we also continued with the E21 placenta, we observed that the trend was still similar, except for CXCR2, which was not upregulated, and then CCR2, which also did not have a correlation at E21. But everything else, apart from also IL-10, these were significantly upregulated. There was a significant correlation at the E21 time point. So having observed this finding in the placenta, we were then interested in finding out what is happening in the brain. So in the prenatal brain, we observed that yes, there was a significant upregulation in the microRNA miR223 in the choreo brain. However, at E21, it appears that this upregulation was attenuating. So we next looked at the various inflammation genes, and there we observed that at E19 time point, only CXCR2, which will be mainly expressed in neutrophils, and also CXCL1, as well as TNF alpha and then IL-6, these were the only upregulated microRNAs. Inflammatory genes. So we're next interested in the correlations between the microRNA miR223 and also these inflammatory genes. We looked at the E21, and here we observed that for the upregulated ones, only CXCR2 was significantly upregulated. So next, trying to find out the correlations, there we observed that only CXCR2, CXCL1, CCR2, and then NLPR3, which were pro-inflammatory genes, had a significant strong positive correlation with the microRNA miR223. The rest did not show any association. So when we looked at the E21, we observed that here again, only CXCR2 was significantly showing a positive correlation, suggesting that as there was more development in the perinatal brain, CXCR2, which indeed literature shows is upstream of the microRNA miR223, this was significantly upregulated and also had a significant strong positive correlation. So we're next interested in what is happening in the postnatal brain in these rats. When we looked at the CXCR2, we observed that yes, this was still upregulated in the P7, which is the human term equivalent of rat. And then we're also interested in another gene, NR2B, which is an NMDA receptor gene, and in the brain, it is mainly produced by excitatory neurons. And here we observe that this was also significantly upregulated, but by day P21, which is equivalent to the young toddlerhood of humans, these two genes, CXCR2 and then NR2B, were no longer upregulated. So we were also interested in the correlation, and then we observed that here again, CXCR2 was exhibiting a strong positive correlation with miR223 at P7. However, there was no correlation between miR223 and then NR2B at P7. However, at the P21, there was no correlation in any of these genes. So having observed this led us to formulate a hypothesis that during chorioamnionitis, CXCR2 was upregulated, and then this leads to upregulation of the microRNA 3, which in the placenta leads to upregulation of all these genes. In the brain, only CXCL1 and then CCR2 were upregulated. We did not observe any change in the expression of the NR2B gene. So by conclusion, we can say that our data indicates that the inflammation elicited microRNA 223 is upregulated in both placenta and brain, and this is as a result of induction of chorioamnionitis. And we also observed various inflammatory genes were upregulated, mainly in the placenta and then in the brain of these subjects subjected to chorioamnionitis. We also observed a strong positive correlation between MIR223 and then the inflammatory gene, mainly in the placenta. And in the perinatal brain, we observed that there was a significant positive correlation between the microRNA MIR223 and then the CXCR2 in the perinatal brain, both at E21 and also at the human term equivalent of P7. So from this data, we can say that studying microRNA expression could also contribute to post-transcriptional rules in conditions of perinatal brain injuries, such as in cerebral palsy, which this model actually is able to establish. And I want to thank my colleagues in Johns Hopkins in the Department of Pediatrics, where this work was done, and also the funding agencies, both from Johns Hopkins and also the NIH. And also thank you all for your kind attention.
Johns Hopkins University.
Dr. Thomford, you're muted.
All right. Thank you, Stephen. So the last person to present for this session is Edward Intim from KNUST, and you'll be presenting on chemophysical characterization of some antimicrobial peptides that will be hinting at intracellular target binding. So Eddie, for the next 15 minutes, the floor is yours. After him, you then do the question time.
All right, so thank you very much. Good afternoon. I'm Edward from KNUST Department of Chemistry. I've been studying antimicrobial peptides from marine invertebrates, Olivancilleria hiatula, and recent findings is what I would like to share with you, especially on the aspects of chemophysical characterization, as well as some sort of in silico studies as to what target this peptide affect in order to elicit its antimicrobial activity. So the age-old problem of antimicrobial resistance in recent times threatens to send us to a post-antibiotic era where most of our antibiotics are not able to kill bacteria and enhance good health and well-being globally. Well, the critical priorities of the World Health Organization, Pseudomonas aeruginosa is one of the multi-resistant bacteria species on the list, which is prevalent in this sort of threat, which surrounds us amidst all other disease states. Could be viral, could be all others. Biofilms are what is predominantly known in bacteria for antibiotic resistance. What are biofilms? Biofilms are shields or covers that bacteria are able to create in order to block the effect of antibiotics and other biocides. Biofilms are created as persistence mechanism as well as a strategy to defeat the host immune system. So it provides some sort of solidarity where pathogenicity is enhanced as well as growth. Because there is this enhanced protection, most bacteria and some viruses and other microbes adopt biofilm formation as their growth strategy. So this shows how bacteria make colonies. After they are in their free states, they make communities. And then from these communities, we have matured biofilms. When resources are meager, bacteria cells get detached to form new colonies and the cycle proceeds. And this affords them enhanced pathogenicity and protection. So there is no known anti-biofilm agent, and research in recent times is looking at how to deal with microbial infections from a multifaceted angle. Antimicrobial peptides have become attractive in the sense that they possess very good antimicrobial activity on biotic and abiotic surface. They have a direct killing effect of bacteria, as well as they are able to prevent biofilm formation. From biofilm formation, they are able to bind to quorum sensing signal and then interfere with bacteria cell-to-cell communication that help them to form biofilms and resist any sort of antibiotic. In cases where biofilms are already formed, peptides are able to penetrate the biofilm, which is usually made of the exopolymeric substance, and kill persistent bacteria cells. Also, they're able to help neutralize endotoxins that are produced by bacteria in enhancements of pathogenesis. So in brief, antimicrobial peptides have broad spectrum activity, and with their multimodal capacity, they serve as a novel approach to biofilm related infection treatment. So where are we going to get peptides? In the literature, peptides are sourced from plants, usually some from insects, some from amphibians. The African toad has proven to be very resourceful in that direction. Aquatic environments have also been explored. And usually the microbial activity is used to classify what sort of peptide you have, be it antibacterial, antifungal, anti-inflammatory, antiviral, antiparasitic, and anti-cancer. So there are a broad spectrum of activity related to antimicrobial peptides reported thus far in the literature. And classification by amino acid composition of these peptides have been revolving around key amino acids such as glycine, tryptophan, histidine, proline, and arginine. And the structure of the peptide too also relates to its biological activity to a large extent. So we sourced our peptide from a marine invertebrate, which is Olivancelaria hiatula from the Western region. And then peptides extracted from this marine invertebrate was subjected to investigations of its antimicrobial potential briefly. We specifically determined the biological activities of the peptide extract, also did some isolation purification studies, and also determined biological activities to a large extent. We also hope to be able to elucidate the potential mode of action of the peptide in silico. Thus far, we've been able to determine the biological activity, the extent of purity by HPLC, and its ability to attenuate quorum sensing in Pseudomonas aeruginosa. We also have this work scheme where we use the sequence of the peptide to elucidate what targets within the bacterial cell these peptides from Olivancelaria hiatula binds with. Thus far, the results we have shows that the peptide extract indeed has biological activities of those in the class of antimicrobial peptides. Based on their preliminary characteristics from infrared spectrometry, we know that they have some level of hydrophobic content as well as alpha helical behavior. Antibacterial activity was very significant, and we tried to do a combination with existing antibiotics, and we realized that there was very good synergy. However, there was antagonism with a macrolide, which we tested, which is erythromycin. But most of the other antibiotics we tested worked in synergy with the antimicrobial peptide. We also went ahead to try to elucidate the mechanism of action of this peptide. However, critically, there is limited information on what this peptide could interfere with in the bacterial cell. So we matched the quorum sensing inhibitory effect, the synergism and antagonism, with that of gentamicin, which is an aminoglycoside with very good anti-quorum sensing effect, as well as the antagonism in the erythromycin guided us to kind of think about what possible targets in the bacterial cell this peptide will be interacting with. And we realized that in the lines of azithromycin, this peptide could be interacting with bacteria ribosome, and thus kind of preventing protein expression that is involved in quorum sensing. So we went ahead to review information available on antimicrobial peptides that are able to interact with bacterial cells. We realized that the bacterial cell membrane was the most preferred target, followed by some intracellular targets such as the DNA, RNA, and then the bacteria ribosome. So we analyzed the purity and realized that the peptides were pure from extraction from our RPH PLC. And then we proceeded to determine the amino acid composition of the peptide. From the visible scan, we realized that there is just little aromatic amino acid composition, which usually absorb around 280. However, the peptide backbone gave us the highest maximum absorbance, which was around 195, and then a lower arm around 243, which is characteristic of peptides indeed. So from the amino acid composition, we realized that proline, alanine, glycine content, glutamic acid contents. When we did a comparative study, we realized that, well, proline content outmatched all of them, followed by the glutamic acid content. So in comparison to what is known in the antimicrobial peptide database, we could guess at the fact that the peptides from Olivacilleria hiatula targets intracellular organelles such as the ribosome or DNA and nucleic acid related substances. We decided to elucidate this in silico. However, we needed the structures of the peptides to be able to do this with the computer. Since the sequence was not available, but we had information on the amino acid composition, we designed peptides which were composed of the amino acid composition of the peptide we extracted from Olivacilleria hiatula. So this design was by sequence shuffling, antimicrobial peptide characteristic prediction, and then direct antimicrobial activity prediction with some online servers. And then with the 5 main hits which we designed, we solved their structures and realized indeed there was some level of alpha content as the infrared spectroscopy displayed. OHPRP5, which is our 5th hit, was similar to a known proline-rich antimicrobial peptide, which was to that effect kind of encouraging to screen in vitro. And in vitro, we realized that these peptides were active with when treated against bacteria. We chose oncosin as the control because it's a known proline-rich antimicrobial peptide, which have been co-crystallized with Thermophilus ribosome, 70S ribosome. So we tried to dock the peptides against the Thermophilus ribosome, and then we realized that it had binding affinity to the regions where oncosin also binds with affinities of about -9 kilocalories per mole, which signifies very strong association. Other binding modes predicted from the molecular docking study show that there are other regions on the bacterial ribosome that these peptides that have been designed from the amino acid composition of Olibacilleria hiatula peptide could bind on the so for that of the third design peptide, we realized a good binding affinity of -9.0. The fifth one, we also observed -9.3, which is in similitude of that of oncosin, which is a control, where we measured a binding affinity of about -10.10, indicating good binding. So in summary, peptides from the marine invertebrate gave us impressive antimicrobial potential, helped to a large extent attenuate biofilm formation, also suppress virulence factor expression in Pseudomonas aeruginosa. We have also shown by our method that the extraction technique yielded pure proline-rich antimicrobial peptides. And with the antimicrobial activity of the design peptide in vitro, we recorded concentrations of about 95 to 279 micromolar as the minimum inhibitory concentration for bacteria. Largely, these peptides give bacteriostatic effect. It means that after 24 hours, these bacteria cells begin to grow after they have been inhibited. They begin to grow on, and this has been the case in literature and elsewhere. We are working actively to determine the sequence of the peptide. We are collaborating with Medina in Spain, where we collaborated after last year's conference. We thank COBREG so much for such a platform. These are some references, and I acknowledge the West African Research Association for supporting this research to a large extent. Thank you also for your attention.
All right, thank you very much, Edward. So we had 4 presenters: Ekuya, Franklin, Stephen, and Edward. It's question time now. If any of us has any questions for any of the presenters, kindly type it in the chat console or raise your hand and tell us the person you are directing the question to, so that the person would answer appropriately. Any questions?
I would have one for Ekuya, if that's okay.
All right.
So Dr. Karakari, what, can you give us any speculation as to what might be going on with thinking in terms of the possible common underlying biology between malaria and neurodegenerative disorders, as you alluded to in your presentation?
Okay. Hello, Prof.
Yes.
So you're asking some of the similar biological processes which may be similar between neurodegenerative diseases and malaria. Is that
malaria? Yeah.
Yes. So I think mainly it's got to do with inflammatory response, and because majority of the genes that were upregulated in malaria were related to inflammation, which is also seen in Alzheimer's disease. So there was this publication that came out that about 70% of the genes that are implicated in Alzheimer's disease have got to do with inflammation and lipid metabolism. And so I think that that is the connection between malaria and Alzheimer's disease with regards to inflammatory process. I don't know whether it's your question is answered.
Yeah, I was just wondering. Thank you. Thank you very much.
Yeah, you're welcome.
Yes. Any other question for any of the presenters? Any other question for any of the presenters? Okay, so Stephen, I have just a question. You know, basically when we are looking at microRNAs, their mechanism of action is to bind to mRNAs, basically degrade them. So ideally, you'll be expecting more of a down regulations more than an up regulation. But we can see that your study, you found a lot of up regulations in other forms of genes that are related to microRNAs. So I was wondering whether couldn't it be as a result of the microRNA 223 rather affecting maybe other genes that interact with these genes that could have probably allowed the upregulation of those genes? I hope you understand my question.
I actively contribute to the solution. So that's on a light note. So to answer the question, indeed, literature shows that microRNA is upstream of the NR2B I mentioned. It is also both, IL-6 is both upstream, it elicits 223. However, it's also a target of. To answer you directly, NF-kappa beta and NLRP3, they are both downstream targets. So, being down regulated, that certainly was. And because this is also, let's remember, this is still the brain. I mean, both the brain realize that at this center, you already obviously have the things. Environment, you have most of this pro-inflammatory genes already elevated. So this perhaps is a case more of correlation, not causality. But CXCR2, for example, is upstream of year 223. So year 223 having that positive correlation, that makes sense. And you observe that was also able to still be observed both at E21 and then P7. So what's to be able to answer your question well, we perhaps may need to continue this at the P21 time point and also we also took people at P60, those would also have to be analyzed to see whether these changes then correlate. In that case, if we see the inverse correlation, which you're expecting, that we can say, and let me just add briefly that for my PhD work, I look at this microRNA and then the neuronal genes, and I can tell you that we had what you are talking about, but that was mainly for psychosis, not cerebral palsy. So yes, maybe prenatally, because the blood-brain barrier has not been developed.
Okay. Questions in the Q&A session. I can't see. I can only see okay. There's Silas will say. Okay, now I see the Silas. All right, Silas, I see your hand up. I think my screen is funny, so I couldn't see the hand up. Silas, you can ask your question. Silas, I think you are muted, so you can unmute. I think this one is a question. Okay, so there is a question for Ekuya where the person, okay, so that's also from Silas. He's asking how she detected malaria in the patients.
Okay, so this is coming from, hello?
Yes, yes.
Okay, so well, this study is based on previous analysis of studies by two groups. So they had a little bit of differences in scoring the patients. So with regards to mild malaria, so the patient had to have fever and parasitemia levels had to be less than 25,000 parasites per microliter of blood. And they shouldn't have any forms of or proofs of severe malaria. So that means the person shouldn't have any hypoxia, respiratory distress, or anemia. For severe malaria, they classified the patients based on hypoxia, hypoglycemia, but without neurological symptoms. And the hemoglobin level should be around less than 5 grams per deciliter. That was for both et al. For cerebral malaria, so obviously the characteristics had to be, they scored with either the Glasgow or the Blantyre coma score, depending on the study. So for Thiam et al, it was the Glasgow score of less than 9. And for both et al, it was a Blantyre coma score of less or equal to 2. And the patient should also had some presence of deep coma or absence of purposeful responses. So that was how they determined or characterized the different forms of malaria. And I hope that answers your question, Silas.
All right. We are, I think, on time for a changeover. So if you have any other questions, you can use the console so that we direct it to the various presenters back door for them to answer those questions. So I think the next moderator is Dr. Winifred Amwakon from the University of Nottingham, UK.
Dr. Amwakon was not able to make it, so Dr. Linda Amwa will be the next moderator. Dr. Amwa, you may take over.
Hello, and thank you very much for staying on for the program. We have 4 more talks, and then we have questions and answers. And just as previous, each talk will be 15 minutes, and we will hold on to the questions till after the 4th talk. We presently have David Ntiamwa Ofosu from the Department of Basic and Applied Biology, University of Energy and Natural Resources, Sunyani. And he will give us a talk on the assessment of APTT results in hemophilia A patients and the impact of COVID-19 on their health, a study at their Konfanochi Teaching Hospital. So at this moment, I would leave this to David to take over. David, we're listening, and you have 15 minutes. Thank you.
Thank you, Linda. Am I going to be given an opportunity to share the screen?
You will.
You should be able to share your screen.
David,
yes.
David, if you entered as a panelist, you will see the share screen.
Okay. Let me find that. Yes, I can see share screen.
Yeah, so load your slides there.
Okay. Okay. Please, can we all see my screen?
Yes, we can.
Thank you very much. My name is David Ntiamwa Ofosu, and this is a study part of an ongoing PhD, which I'm currently doing at the Department of Molecular Medicine in KNUST. And we found that the hemophilia patients that were in Konfanochi, who were being monitored actually before the COVID came in, after the COVID, we found that there were a few issues that we wanted to go in and try and find. So we decided to assess the APTT status and look at the impact of COVID-19 on their health. So I did this study with my colleagues, Prince Kodom, Emmanuel Samuel, and Dote, who are all from Garden City. So hemophilia is an X-linked disorder which results from genetic mutation of people who are having some changes in their factor VIII gene. And the factor VIII gene is very important in the production of certain factors that help to stop bleeding when we have a cut in our body. Now, the severity is dependent on how much of the factor VIII gene a person can make. And hemophilia is usually associated and shown in patients who are males rather than females. So here they would usually have bleeding into their joints, and they'll have bleeding into the skin, as we can see from the pictures here. And it makes the knee and the joints in their body difficult to control. And so these patients would usually need prophylactic treatment. Sometimes they have to come for treatment about 4 times in a month, or at least once in a month at the hemophilia clinic. So there are currently over 120 hemophilia patients in the registry in Kofanoche. We have some registry in Accra, but I didn't look at them currently. And the challenges that they are having with their care recently has been highlighted, especially when the COVID came, because they had to come almost every other week to be given external factor concentrate for the body to be able to manage bleeding tendencies in their body. And when the COVID-19 set in somewhere late 2019 and the greater part of 2020, most of these patients were told not to come to the hospital facility. Most of them too were afraid that they were going to catch COVID. So despite the challenges that they had with managing their bleeding tendencies, they were still not able to come to the hospital. And so the World Federation of Hemophilia had to issue specific recommendations for how these patients should be managed globally, including the hygiene measures, including information for how they can be managed if they have exposure to COVID, and how they should be assessed whilst they are at home. But unfortunately, in Ghana here, we realize that because there isn't much education, there isn't much reach, there isn't much research, and even capacity to treat, managing these patients has become a challenge. And COVID-19 came to even complicate the issues that they had. But we are unable to know the specific impacts that had on these hemophilia patients when we went particularly into the lockdown and were prevented not to come out and things like that. So one of the greatest challenge that we had, which we believe is a justification for this work, is that most of the factor concentrates were donations that were given to Kofanoche Hospital or through the Ghana Hemophilia Society. And because of the lockdown and the close of borders in many of these countries, we realized that there was shortage of replacement products like the factor VIII concentrate. So even if they decided to come to the facility, they were not going to get them when they came. So that became a challenge. So we decided to assess what exact impact COVID-19 had on their lifestyle and also on their bleeding tendency. And we were going to do this by measuring their APTT levels. APTT is a test that can be done in any patient, but people who are having hemophilia, the APTT test is going to give us a prolonged result, which tells us that when they have a cut, they are going to bleed for a longer time. And sometimes if we do not apply external force, they will bleed till some even die out of the bleeding for a longer time. So we decided to determine their APTT results before the COVID-19 pandemic set in. Then we were now going to determine their APTT results after the pandemic. So we had started this study through my PhD, where we had determined some of their APTT results already. So we decided to now measure their APTT results after the pandemic and compare, and to also determine if there were any factors that may contribute to the changes in the APTT results and the ratios, and to also find out if there are any challenges that were faced by these patients after the pandemic. So we were able to recruit about 44 participants into the study. They were all males and they were all on the hemophilia registry already. Patients who had other bleeding disorders like von Willebrand disease were excluded from the study. So in the test, we performed the APTT. We took retrospective data for their results in 2019, and we also ran their tests ourselves in 2021 and then later part of 2020. And we were able to determine their APTT ratio. And we used the reference that was determined by Ofosu et al. Paper we authored in 2019, where we determined the reference range, which was 26.3 to 44.1. So that's what we used in this study as the reference interval to study our patients. And based on that, we took the normal APTT ratio for the study to be 1. So if a healthy person should have APTT ratio of 1 or a little above 1. So in our results, the range of patients we took were between 2 years. The minimum was 2 years and the maximum age was 27 years. And these patients, they are people who are having bleeding. They were having bleeding tendencies in terms of hours, about 30 minutes to about 360 hours, 330 hours in a month. So the minimum bleeding time was 30 minutes and the maximum bleeding time was about 330, more than 330 hours in a month in these patients, with average having about 85 hours bleeding in every month. Now we realize that their pre-APTT results before the pandemic, the mean was about 70 seconds. So the mean for the hemophilia patients was about 70 seconds bleeding, and comparing it to the upper limit for APTT, which we wrote here, was 44. That means that these people who were having 70 were having prolonged bleeding. Now, after the pandemic, when we undertook the study, realized that the APTT, the mean shot up to 176 seconds. That is close to about 3 times the bleeding. And it means that the pandemic was going to cause them to have more bleeding. And the difference in the APTT ratio, 70.2, and the post-APTT of 176, this difference was statistically significant. We also found that their APTT ratio was 2.1, pre-APTT ratio before the pandemic, which also shot up to 5.5. So they were going to bleed about 3 times more after the pandemic. And that difference was also statistically significant. So this figure here was to demonstrate the distribution of APTT results before the pandemic and distribution of APTT results after the pandemic. You realize that results in this table, the mean values were higher and above the mean values in the pre-APTT results that we saw. Again, we tried to find out how many times they visited the hospital before the pandemic set in. And you can see from this pie chart that about close to 70% visited the hospital 4 times in a month, which means they were coming there every other week, every week. About 70% were coming there every week, and about over 22% were coming there twice in a month. The least that came once in a month was about 7% of the hemophilia population. But during the lockdown and after the lockdown, as much as 70% are staying home because they were afraid or for other reasons, they were not coming to the hospital. And this definitely may have contributed to the increased bleeding tendency that have been exposed to these hemophilia patients. And so in this table, we have already described how many were coming to the hospital and how many have come to the hospital after the pandemic. what was interesting to us is that when they did not come to the hospital, as much as 24% were trying to use local herbs to be able to manage hemophilia. Maybe we should go to the patients, something that was interesting. We are going to go to the patients and to find out what exact local herbs they were trying to use. But I want to believe it hadn't worked too well. But this is a source of worry, about 24%, that is about a quarter of the population are now trying to use local herbs to be able to manage their bleeding because they were afraid to come to the hospital. So in our discussion, we said that inherited bleeding disorders have become, it's a rare disease, and we need to have specific management, specific protocols for them. And before the pandemic, the Ghana Hemophilia Society used to go around and have training for physicians. Now, when the pandemic set in, managing these patients who in the midst of restrictions have become difficult. Obviously, the clinician is afraid to have the close handling or close management of these hemophilias. And most of these hemophilias, which unfortunately are coming from homes that are not so well to do. In one of our studies, are not literate enough to be able to get confidence to come to their facilities for the fear of catching COVID. So most of them will not come to the facility. Most of them think that when they come, there is no concentrates to give them. And the challenge, one biggest challenge was that they were not even given any information. They didn't have, like in other countries where Hermans et al. And Bentop et al. Reported that when there's a pandemic and there were special people with these needs like hemophilia, the facilities were in touch with them at their homes. They called them, interact with them. Here, measures like this were not taken by the Ghana Service. And so we just left our patients to their own fate, and it has rather increased their bleeding tendency, which could adversely affect their lives and complicate the issues that they are in. So in conclusion, the fear of COVID-19 have left many of our patients to be suffering. And they are suffering because they are avoiding to come to the hospital. And that is going to make their eventual management even more difficult. And so we believe from our study that the healthcare providers, especially the hemophilia nurses, the hemophilia doctors, should, as a matter of urgency, be able to contact the hemophilia patients who are on their registry and give them supporting management, give them the assurances that yes, COVID-19 is not gone, yes, but there's a place for them to be able to come to the hospital facilities so that we can be able to take good care of them. So this is what we find. These are a few references that we used for our study. I'm very grateful for listening. Thank you very much.
Thank you very much, David. You were just on time. Thank you so much for your talk. And for all those who have questions, please hold on. And at the end of the session, you have an opportunity to ask your questions. So at this moment, we'd like to welcome Elizabeth Aban to talk. And Elizabeth is from the Department of Medical Diagnostics, Kwame Nkrumah University of Science and Technology in Kumasi. And her talk is entitled The Prevalence of Bleeding Episodes and von Willebrand Disease Among Female Students in the Kumasi Metropolis, Ghana, a cross-sectional study in 2 selected universities. Elizabeth, you're welcome.
Okay, thank you very much. Please, is the slides showing?
Yes.
Hello. Okay.
Yes. We are seeing your screen, not your slides.
You need
Not
to
the
open the
slides. Okay. I'm sharing again. Is it visible now?
Yes.
Okay. So my name is Elizabeth Aban. I'm presenting on my research on bleeding episodes and von Willebrand disease in female students, a study in 2 selected tertiary institutions in Ashanti region, Ghana. So inherited bleeding disorders are a group of coagulation disorders characterized by a wide range of bleeding symptoms with variable inheritance patterns. And von Willebrand disease is the most common autosomal inherited bleeding disorder caused by either a deficiency or an abnormality or a virtually absent von Willebrand factor. So what is von Willebrand factor and its role in hemostasis? Von Willebrand factor is an adhesive protein that promotes platelets adhesion and aggregation to the site of a vascular injury to form a primary platelet plaque in order to stop bleeding. It is also a carrier molecule for clotting factor 8, which protects it from being degraded or cleared from circulation. And so when there are normal levels of functional von Willebrand factor, the role of factor 8 in the coagulation cascade is made possible. And so fibrin is formed in the end to stabilize the primary platelets plaque that has already been formed. Globally, the prevalence rate of von Willebrand disease is 1 to 3%, with a high prevalence of 5 to 20% in females with menorrhagia. Now, due to the autosomal inheritance pattern of the disease, both males and females are equally affected. But bleeding episodes are seen more frequently in females due to menorrhagia and childbirth. So bleeding episodes over here refers to any mucocutaneous bleeding, such as gum bleeding, nose bleeding, and the like. So the problem in Ghana has been that many females usually report to the clinician with complaints of heavy menstruation and other bleeding episodes. Some may not report this at all, as they may attribute this to history of heavy menstruation or other bleeding among females in their families. And so those who report this are usually asked if they have a family history of bleeding or if they are pregnant. And so the common routine tests that are often performed are full blood counts and then urinary pregnancy tests. And then in few cases, a pelvic ultrasound scan might be performed, and it ends there. Not forgetting that these bleeding episodes may be caused by an underlying inherited bleeding disorder, mostly the mild forms, which are often left undiagnosed. And so the consequence of this bleeding is the many complications associated with anemia, iron deficiency anemia being the most reported in these instances. Now, one major problem in Ghana is that not much attention is given to these bleeding episodes and their underlying factors. And also von Willebrand disease testing is not done in Ghana routinely. And till date, not a single study has been performed or conducted in Ghana. And so the study sought to determine the common types of bleeding episodes and the prevalence of von Willebrand disease and associated risk factors among females in these institutions. And to achieve this aim, the most common types of bleeding episodes were determined. The frequency of von Willebrand disease was also determined. An association between von Willebrand disease and bleeding episodes was determined. And the study also determined hemoglobin level, RBC indices, and prothrombin time and activated partial thromboplastin time in persons with normal and abnormal von Willebrand disease status. Von Willebrand factor activity and antigen levels were also determined in relation to ABO blood group. And lastly, an association of clinical characteristics with normal and abnormal von Willebrand disease status was determined. So the study was a cross-sectional one conducted in KNUST and the Garden City University College between December 2018 and June 2019. 75 apparently healthy Ghanaian female students were recruited from each institution. They were within the age range of 18 to 35 years, excluding those who were pregnant, breastfeeding, and then menstruating, and also on certain medications such as aspirin. Ethical clearance was sought, and then questionnaires administered to obtain social demographic data and bleeding history. Pictorial blood loss assessment charts was also administered to determine menorrhagia. Full blood counts was done after blood samples had been collected. ABO, using the SysMS automated hematology analyzer, ABO blood grouping was also done using ABO-anti sera in a standard tube method. PT and APTT were done manually, and then vulnerable factor antigen and activity assays were done using an automated analyzer manufactured by the instrumentation laboratory. Data was analyzed using R package. So table 1 shows the baseline characteristics of the study population. More than half of the participants were within the age range of 18 to 24 years, with most of them in their second year, and a greater proportion of them were blood group O individuals. Table 2 confirms the specific objective 1, where common types of bleeding episodes were determined. So bleeding episodes was presented in 42% of the participants, with the most common being menorrhagia. The 42% prevalence was similar to a postulate by Sadler, who reported that about 46% of females will have at least one bleeding episode. However, this 42% prevalence was not in line with a study by Freiberg et al., who also conducted a similar cross-sectional study in females in Sweden. They found a prevalence of 73%. Now, the 38% prevalence of menorrhagia in our study was similar to that of Freiberg. They found a prevalence of 37%. Figure 1 shows the prevalence of von Willebrand disease among the entire study participants. So 7 represented 4.7% had von Willebrand disease. And this was similar to a study by Gezel et al., who also conducted a similar study in Turkey. They found a prevalence of 6.5%. And this finding is also similar to a report by Chen et al., who reported that the prevalence of female von Willebrand disease in females is twice that known in males. Now, all the participants who had von Willebrand disease were those with type 1 von Willebrand disease, and this is similar to a study by Lillicrap and James. They found about 75 to 80% of their participants to have type 1 von Willebrand disease. And the 100% prevalence of type 1 von Willebrand disease in a study could mean that probably at the time of the recruitment, only type 1 von Willebrand disease individuals participated in the study. Table 3 shows an association between von Willebrand disease and bleeding episodes. So out of the 7 who had von Willebrand disease, 7.9% of them had bleeding episodes. There was, however, no association between bleeding episode and von Willebrand disease. Again, those who were at risk of having von Willebrand disease, in other words, low von Willebrand factor, 12.7% of them had bleeding episode. And this is not in line with Munla et al. Study, who found 38.8% prevalence. So figure 2 compares hemoglobin and Russell indices between the participants with normal and abnormal von Willebrand disease status. So von Willebrand disease status over here refers to the presence of von Willebrand disease or low von Willebrand factor. And then the blue violin box plot over here refers to those who are normal for von Willebrand disease status, and the red represents abnormal. So MCV and MCH were significantly high in those who were abnormal, even though their individual values were within normal range. This could be explained by the fact that probably these individuals might have experienced some form of blood loss. And so this has been compensated for through reticulocytosis. And this was similar to a study by Exbo et al. Now, even though the hemoglobin level was low in those who had abnormal von Willebrand disease status, again, their individual values were within normal range. And so this was not so representative of anemia. This was statistically insignificant. Now, figure 3 compares PT and APTT between the participants with normal and abnormal von Willebrand disease status. So PT was normal in all our participants. This was statistically insignificant. However, APTT was significantly high in those who were abnormal, and similar to a study by Sheriff et al. again, their individual values were within normal ranges. So it could be due to the fact that probably their factor 8 levels were high enough to have caused a normalization in their APTT. Figure 4 shows the levels and activity of vulnerable factor stratified by blood group. So there was no significant association between vulnerable factor activity and antigen in relation to ABO blood group. However, there was an increase in the vulnerable factor activity and antigen in persons who were AB, the AB blood group individuals, with a decrease in the O blood group individuals. Reason being that there is expression of ABH antigenic determinants on vulnerable factor. And so obviously AB individuals who possess A and B antigens will have a full protection, because what the antigenic determinants do is to protect the vulnerable factor from being degraded and cleared from the circulation. And so they will have a full protection as compared to the O individuals who do not possess the A and B antigens. So from the previous figure, this table is confirming the fact that all individuals have got 3 times increased risk of having abnormal von Willebrand disease status, and this was statistically significant. So in conclusion, bleeding episodes were greatly manifested, with the most prevalent being menorrhagia. Von Willebrand disease is a common inherited bleeding disorder among female students in Ghana. And it is important to note that not all bleeding episodes are caused by von Willebrand disease. People with von Willebrand disease may not manifest anemia, and normal APTT does not rule out von Willebrand disease. Lastly, blood group O individuals are at increased risk of having abnormal von Willebrand disease. And so it is recommended that females who report with complaints of bleeding are screened for hemostatic disorders, especially von Willebrand disease. And also a broader study involving a larger sample size should be looked at, focusing on inherited bleeding disorders. So I'm grateful to my supervisors, Professor Otriade Mensah, Dr. Enoch Odame Anto, and then Mr. David Ntiamwa Ofusu. I'm also grateful to Dr. Yabuama for the betray of Garden City University College, and also to all the staff of the Department of Hematology, Confanoche. These are some references. Thank you.
Thank you very much, Elizabeth, for the very nice talk. We will hold our questions until later, and thank you so much for making the time. We now call on Lucy Bawine from the Department of Medical Diagnostics, Faculty of Allied Health, Kwame Nkrumah University of Science and Technology in Kumasi, to give us a talk on differential assessment between iron deficiency and alpha thalassemia among microtic hypochromic anemia individuals in the Upper East region of Ghana. Lucy, you have 15 minutes.
All right, thank you. Thank you very much. Sharing my slides. Please, if Dr. Amfo is there, can you help me to share the slides? I'm having a little challenge.
Do you see the share screen, the green icon in the middle of the bottom?
Yes,
of the
please.
screen? Click on it. If you click on it, then you can click on the particular screen that you want to be shared. Have you tried it? Lucy, how's it going? Okay. Okay, there you are.
Hello, please, can you see my slides?
Yes, we can see them. You can go on.
Are my slides visible?
Lucy, you can go on. We can see them.
All right, thank you very much.
Mm-hmm.
My name is Lucy Barini, presenting on differential assessment between iron deficiency and alpha thalassemia among macrocytic hypochromic anemia individuals in the Upper East region of Ghana. Anemia is the most reported red cell disorders in most developing countries, with high prevalence in pregnant women and children. It is reported that 60% of African children are anemic. Macrocytic hypochromic anemia, one of the classes of anemia, is more prevalent among anemia cases. This condition is an abnormality in full blood count, which prompt clinicians to do further investigations for the cause, which include iron deficiency, alpha thalassemia, anemia of chronic diseases, sideroblastic anemia, and occasionally lead poisoning. Macrocytic hypochromic anemia result from deficient hemoglobin synthesis in the red cell, causing both MCV and MCH to reduce. The degree of the macrocytosis is directly proportional to the number of alpha genes that are deleted, ranging from a single gene deletion with no or mild microcytosis to an intrauterine death where all 4 alpha genes are deleted. In Ghana, data on alpha thalassemia is very scanty. Studies, however, have shown that 26 to 33% of the general population are carriers for alpha thalassemia. Macrocytic hypochromic anemia, however, is mostly associated with iron deficiency in our settings. Therefore, alpha thalassemia is most frequently mistaken for iron deficiency. Hence, differentiating iron deficiency from alpha thalassemia becomes very necessary in order to avoid administration of excessive iron supplement to individuals who are already at risk of iron overload. In order to do this, the aim of our study is to determine the difference between iron deficiency and alpha thalassemia in macrocytic hypochromic anemia individuals for effective management of anemia. And our objectives were the estimation of red cell indices and serum ferritin among our study populations. Then we also determined C-reactive protein status of the study participants, and also determine the prevalence of alpha thalassemia among our participants, and also differentiated microcytosis due to iron deficiency from microcytosis due to alpha thalassemia. This study was a cross-sectional study carried out at the Regional Hospital, Bolgatanga in the Upper East region between November 2018 to May 2019. It included 154 participants and 20 apparently healthy individuals as controls. Macrocytic hypochromic anemia cases were our inclusion criteria. Was patients who were patients with chronic diseases, pregnant women, and patients on iron supplement were excluded from the study. Ethical clearance was sought from the university's ethics committee, and data was obtained from full blood count using Sysme's machine. Then iron studies was done, basically serum ferritin, where we use ELISA as the method for determination. Then we determine C-reactive protein in the participant using quantitative test. and also PCR was done. The data was analyzed using SPSS version 22. Table 1 indicates the demography of our study participants, where we have 49 cases of the participants being males, and 106 of them were females. Also, we have 59 as children that were less than 5 years of age, and then 96 as 5 years and above. Table 2 indicates red cell indices and serum ferritin among our study participants and then the control group. From the study, we noticed that from here, we realized that the mean hemoglobin concentration for the participant was 9.5 as compared to the controls. And then the mean serum ferritin was 18.6 for the patient group and 22.9 for the control group. This difference could be as a result of alpha thalassemia patient being part of the patient category as compared to the controls, who were all apparently healthy individuals. Table 3, iron body stores and C-reactive protein status of the study participant. From here, we notice that 50% of our study participants were iron deficient. And we also had 53 of our study participants being positive for C-reactive protein. And this number was excluded from the C-reactive, it was excluded from the PCR process, with the notion that both C-reactive protein and serum ferritin are acute phase proteins and will cause serum ferritin to acute phase protein and will increase in the phase of inflammation. So once C-reactive protein is positive, it assumes that even if the serum protein is, even if the serum ferritin is normal, it could be as a normal or increase, which is also an indication for alpha thalassemia. It could be as a result of an inflammation. Therefore, they were excluded from the study. This finding was in agreement with a study that was done in 2016, which indicated 49% of their participants were iron deficient. However, not consistent with another study that was done in 2018, which gave an 18.6%. The prevalence of alpha thalassemia among our study population was 65.4%, which was in agreement with a study that was done in 2018, indicating a 66%, whilst disagreeing with Franklin et al., which indicated a 52.5%. This is an agarose gel electrophoresis that was done after the PCR, after the extraction of DNA and the PCR process. This was what we did, and sample M represents the DNA ladder, whilst S1 to S11 represents some of the samples. Then if there's a band on both 1,800 base pair and 2,000 base pair, it means that there is a deletion. There's the potential that the person is alpha thalassemia. But when the band is on only 1,800 base pair, it means that there's no deletion. This one is a slide from, it's an image from the peripheral blood smear, where we have a lot of target cells and hypochromic cells, which indicates thalassemia. Table 5 also indicates the hematological profile of iron deficiency with coexistence of alpha thalassemia and then alpha thalassemia patients as compared to the control group. From here, we notice that the hematological parameters were statistically significant between the between the patient group and the control group. But we however noticed that the hemoglobin concentration, the mean hemoglobin concentration for the alpha thalassemia group was 9.8 as compared to a study that was done in 2015, which indicated that the mean hemoglobin concentration for alpha thalassemia patients was 9.3. We also noticed that the mean serum ferritin levels for the alpha thalassemia patient was within normal ranges, which is the 25.1. This ferritin being within the normal ranges could be as a result of this confirms literature that says that even patients who have alpha thalassemia still have normal serum ferritin levels. Therefore, any additional iron supplement to them might put them at risk of iron overload. Table 6 indicates the causes of the microcytic hypochromic anemia that we identified in this study. From this study, we noticed that the prevalence of alpha thalassemia, that is true. We however noticed that both iron deficiency and alpha thalassemia prevalence stood at 38.6%. This, however, does not coincide with a study that was done in 2014, which indicated that the coexistence of iron deficiency and alpha thalassemia was 31.0%. In conclusion, we say persons with alpha thalassemia have normal ferritin levels. Therefore, further administration of iron to such people may put them at risk of iron overload. In our study, we also find out that 65.4% of microcytic hypochromic anemia persons in the Upper East region are carriers for alpha thalassemia. Differentiating between the causes of microcytosis therefore becomes very necessary, not just for appropriate management, but also for appropriate counseling of individuals that are already at risk of iron overload. We recommend that with such high levels of alpha thalassemia reported in this study, steps should be taken to look at microcytosis, to look at microcytic patients, not just as iron deficient patients, but also to look at them as potential carriers of alpha thalassemia. We are, however, limited by sample size as compared to the other studies that were done. Our sample size was low, and also we couldn't determine hemoglobin A2 so that we could establish the presence or the existence of beta thalassemia, and therefore recommend that any further study should include this. These are my references, and I would like to say thank you to my supervisors and all the lecturers at the Medical Diagnostic KNUST. I'm grateful. Thank you very much.
Linda, you are muted.
Sorry. Thank you so much, Lucy, for your talk. It's interesting to know that we don't do too much on thalassemias. At this point, we'll call on Hannah Abankwa to tell us about pediatric triaging, the perception, attitude, and practices among nurses at Weija Bawe Municipal Hospital. Hannah is from the Department of Pediatric Nursing at the Asha Polyclinic in Ghana. So Hannah, you have 15 minutes and we're ready to listen to you. Thank you.
Good afternoon, Lucy. Can you, okay. Lucy, can you kindly share your screen? Share your screen so that I share mine.
Good.
Hello, everyone. Good afternoon to us all. I think today we've heard a lot about microbiology, but we are going to hear a bit about clinical work. So my name is Hannah Banko, as Lucy said, and I'm a pediatric nurse specialist at Osha Hospital. We are no longer a polyclinic, we are a hospital now. And my topic is going to be on pediatric triaging, the perception, attitude, and practice among nurses at Weija Bawe Municipal Hospital. Okay, so our presentation outline is going to start from introduction, the main objective, the method, the results, and discussion, and wait the conclusion. Okay, so one may ask, what is triaging? Triaging basically is about sorting, just sorting out things, either your items or human being. So triaging was stepped up way from the 12th century, but it was actually linked up with a military surgeon who uses this practice to sort out patients in the military field in order to give prompt attention to them. Triaging has gone through a lot of transitions, transformation, and currently it's used as a standard practice in our clinical setting. So triaging helps prescribers or clinicians to sort out patients immediately as they come to the hospital in terms of whether it's a cold case or clinical case. Triaging also helps us to prevent congestion and give us immediate attention to our patient. As the picture depicts, you see that we have an overcrowding hospital where children are in a lethargic situation and they are waiting their turn to be seen. So triaging actually is quite helpful to the pediatric population because during their staging, the anatomical and the physiological, they go through a lot of physiological changes. So we need to make sure nurses that are handling this kind of patients are actually incompetent and they can handle them in order to bring out a good result. So one will ask, what is happening at Weija Municipal Hospital? So the problem out there is they are getting a lot of pediatric clients. As you could see, in 2019, we had about 9,998 pediatric patients who attended the clinic. And out of this, 681 patients, pediatric children, were rushed to the ward and the emergency unit without going through a triaging protocol, which is actually worrying because a child comes to your clinic, you just rush the patient without even going through the normal routines of checking the basic vitals. All that you do is to push them to the ward. What happens? It brings about a lot of congestions and delays in actually picking those with the red flags for us to manage them with emergencies and treat them and to let them go. So our study objective. So with the study objective, it was basically to assess the quality of triaging of pediatric patients by nurses at the Weija Bawe Municipal Hospital. With a specific objective was to assess these nurses' perceptions towards the pediatric triaging, to identify their attitude of nurses on pediatric triaging, to determine the relationship between nurses' work experiences and practices of pediatric triaging, and also to determine the relationship between nursing qualification and practices of pediatric triaging. So the methods, how did I able to come out with everything? The research was a quantitative one and a descriptive cross-sectional study design was employed. This actually should help the researchers to understand the phenomenon at a point in time. Before conducting this research, we had a ethical clearance from both the Ghana Health Service. They went through everything and they gave us the report to start. Also, I had a clearance from the college, the Ghana College of Nurses and Midwives. So we gave it to us. And a formal approval was also sought from the Wage-Agbawo Municipal Hospital, which they gave me the authorization to start. So the participants the population were for the pediatric nurses. Where is the pediatric nurses? These are nurses who actually have influence in taking care of these children directly. So being from the community health nurses, the midwife, the general nurses, they were all summed up to a population of 85. I sampled them using a convenient sampling technique and 70 participants were actually used. But at the end of the day, based on these schedule and disappointments, I had 67 responses for the data analysis. My inclusion criteria included nurses who had worked more than a year with childcare, and also I excluded permanent nurses who have worked less than a year and rotational nurses. The questionnaires, before giving it to them, an informed consent was sought. We explained to our participants, they signed before we started actually working with them. The questionnaires were structured questionnaire and they were given to our participants either in hard copy or soft copy. And Pearson correlation and it was actually, I used the descriptive and inferential statistics to analyze the data set. So with the result, a little background of my respondent. As I said, these were nurses who had in contact with the pediatric population. So one was the educational background was about those nurses with certificate, diploma, degree, and master's. So with the certificate, we had 15 were nurses, and we sum up to 22.4% of the total population. The diploma nurses were 34, the degree holders were 16, and I had one nurse who had masters, summing up to the 66. With the professional background, we had enrolled nurses, perioperative nurse. I had one perioperative nurse. I included this nurse because she works at the theater, and we had children coming with asphyxia and other critical condition from the theater. So I wanted to assess how ready they are to charge this kind of unit. And also we have our community health nurses where children actually come to the weighing. Some come in a critical area, but because they're not able to triage them, they made them sleep through, and then later they come in. We have our midwives, our registered nurse, and our SRN. So that is our old nurses, even though they are registered nurses, we call them SRN, and one critical care nurse. So with the result, one of our variable, as Idea said, was on perception. So with the perception, one of our questions was, is pediatric triaging done at this hospital? I wanted to find out what at all they were doing the pediatric triaging. So in total, I realized that about 80% of nurses strongly agree that triaging was actually being done at the hospital. And we had about 9% disagreeing that they were not doing it, or they were in doubt of something. Also, we wanted to find out whether they needed a mandatory formal training, rather maybe a rigorous training, them getting a lot of formal training on triaging. With that, we had 80% of them this time around also agreeing strongly that they needed it. Even though they were practicing, they still needed more. And we had about 4% who disagree that it's not really quite of a necessity for them to be given that sort of training. And our next question was on pediatric triage, whether it should be for some category of nurses or it should be for everyone. And with that, we had just 35% of them who strongly agree that we should actually be for some few people. And we had about 26% of them agreeing that they disagree. They think that it's supposed to be for the masses. So I think majority of them agree that we should give them regular training for some group of people. Then the last Bible was training is necessary for all nurses. Initially, the first one was on few nurses. This time around, we wanted to find out whether it's supposed to be for all nurses. And with that, you realize that majority of them agree that it's supposed to be for everybody. So triaging for kids should not supposed to be for one particular or a cadre of nurses, but they agree that it's supposed to be for the masses. With all this, this result was inconsistent with these studies. They didn't agree that nurses had poor perception. From the study, realized that our nurses have a good mean score of 71.4%, and they agree that they have good perception towards pediatric triaging, which was quite inconsistent with other studies. Also, in terms of nurses' attitude towards pediatric triaging, we had one, nurses at the place should be put at advantageous position. So we wanted to find, even in this time of COVID, we are putting nurses everywhere. So we wanted to see whether we could triage these children at advantage points. And we had more than half of nurses agreeing strongly that we should put nurses at advantageous point for triaging. And we had few of them, about
Hello?
we shouldn't put them at advantageous point. And we wanted to see whether nurses actually triage these kids at the hospital as soon as they arrived at the hospital. Indeed, a majority of them, about 78%, agree strongly that they were triaging the children. So we didn't know what the problem states were, how they were rushing them to the hospital, but of course, majority of them agree the fact that they were triaging. Fewer of them, about 10%, strongly disagree that they were actually doing that kind of practice. So with the confidence to perform triaging, there's one thing doing it and another thing having the confidence to do it. So we wanted to see whether they have the attitude of confidence. So with this, we had about 75% of them giving as like they agree strongly to the fact that they have the confidence to triage. And we had about 9% of them disagreeing that they had the confidence to triage. And so in summary, we say that the nurses had good attitude towards pediatric triage with a positive score of 84.2%. This findings actually agreed with other studies. Actually, it's a recent study from Afayao et al. In 2017, who found that nurses actually have high positive attitude towards triaging. So in terms of practice, we wanted to see their practices. And one of the questions we asked was, has the hospital adopt colors of triaging a child? And about 60% agree that they actually had a standard coding for triaging the child. And 10% of them disagree that there was actually something like that available for them to triage. In pediatric triage to be done, we also wanted to find whether it's actually done at this hospital. Then about 60% or more agree strongly that it was done at the hospital. A few of them, that's about 7%, disagree that this was done at the hospital. Protocols to triage a child in the hospital. We wanted to assess the availability of standard protocols to triage these children. And more than 80% it's also agreed that there was a protocol in place for them to use as a tool to triage these children. And about 4% of them disagree that they don't have that such standards to triage. And with this, we, after everything, realized that nurses had good attitude to practice, or they're able to practice pediatric triage with a positive score of 57%, which is adequate. So as I said in my methodology, we use both correlation and descriptive. So with the correlation, we wanted to find the relationship. So we realized that nurses had good perception and attitude and practice. There's a relationship between that. So we realize that with all this, these variables, them having a good perception and attitude and practice, nurses are actually going to practice the pediatric triaging better. And you see that there's a significant correlation where it's highlighted. So as well as we're finding out about the perception and attitude, with the relationship, you also wanted to find out about their work experiences. So this variable actually wanted to find out whether a nurse who have worked more could practice, could have a good practice with triaging skills or less. So per this, realized that it was not strongly related, it was not really a significant tool, because there was no any correlation between your work experience and practice. So basically, the research is showing us that if a nurse has a good perception, a good attitude, and towards triaging the children, they can do it better in relation to what your work experience. There's no any significant relationship between your work experience and the practicing of pediatric triaging. And this actually was agreed with this study, considering et al. 2007, who reviewed 4 studies and find out that once you're able to give your nurses knowledge and they have the right skills and attitude towards it, they are going to practice the triaging better, and it doesn't determine their work experience. So in conclusion, most nurses have good perception and practice of pediatric triaging, and we are recommending that there should be regular workshop on triaging for nurses to help them improve on the attitude. Also, the triaging protocol should be integrated into the nursing training. Triaging, actually, you said your work experience doesn't determine how you triage, but when you are given the right tool, the right workshop, the right training, you can do a better job. So we encourage our institution right from the beginning, they should incorporate more triaging for our nurses to be able to do a better job with the children. Also, we want further research to be done. There were limited data in regards to children. So we're encouraging people to go more into a clinical field and do more on the children, and especially on the triaging areas. So acknowledgement, acknowledge my supervisor, Madam Joyce Frawa, who is a lecturer at University of Ghana. Dr. Edubon Safo, who has been an instrumental tool in this work, is a senior lecturer at University of Ghana Medical School, and it's also a co-author for the work. And also Manasi, who also play a role as an author in this research. He is at Ghana Baptist University College of Ghana. And also listening to me, a shout out to you, and all the staff of Weja Bawe Municipal Hospital. Thank you very much. These are my references.
Okay, thank you very much, Hannah, for the nice talk. And so at this moment, I'd like to thank all our 4 speakers and then ask them to all get ready in case there are questions posed for them. For the audience and for those listening, please, if you have a question, can you raise your hand? And then for those who want, they can also put their questions in the chat window, and then I can read those out. So first of all, I would invite people to raise their hands and ask questions. Oh, okay, I have one hand up already. So Dr. Inyamfo. I can unmute you, and then you can ask your question. You tell us who it's directed to, and then they would respond. Thank you.
So one question for Elizabeth and then one for Lucy. So Elizabeth, you found a high percentage in blood group O. Did you follow up to find out why they in particular had a high percentage? And then the second part is looking at your sample size. Is this something, and your results, is this something that you always recommend that is done routinely? Because I thought the sample size was too small and the significance was not too much. And then for Lucy, I want to find out if you followed up on any patients that you thought had been given iron overload. Thank you.
Can you please hear me?
It's shaky.
Hello.
Elizabeth, you have an echo.
I'm very sorry for the noise coming from the background. In fact, I can't control it at the moment, so please bear with me. But I hope you can hear me. Can I go on, please?
Please, you go on.
Okay. So thank you, Dr. Nyanko. With the Black group O individuals, studies have shown that these persons have got low von Willebrand factor, and that is confirmed by the decrease in the von Willebrand factor activity and antigen levels in these individuals from the study. And so, as I said earlier on, there is an expression of ABH antigenic determinants on von Willebrand factor, which protects the von Willebrand factor from being cleared or degraded from the plasma. And so, yes, thank you very much. And so, obviously, all blood group individuals do not possess A and B antigens. And so, they are going to have kind of a lesser protection when it comes to the degrading and then this, what we call it, being cleared from the plasma. Now, with the sample size, after calculating for the sample size, the study found 30. The sample size was actually 30, but then the study adjusted it to 150 sample. And so it was recommended that a larger study involving, yes, a larger study should be looked at in future so as to accurately determine the disease burden of von Willebrand disease in the country. So the sample size was actually 30, but then we adjusted it to 150. So probably in future studies, we are going to look at a much larger sample size. Thank you.
Akwesi, is that okay?
I have to accept the answer.
Thank you very much.
So to Lucy.
Lucy.
Hey, my brother.
Is Lucy here? Okay, as we wait for Lucy, I think I'll ask Stephen to
Well, my question is also to Lucy, so I don't know if it's useful to ask. I mean, but I don't know if any other person has some ideas. It's about the alpha thalassemia. Is there any effect of its expression with sickle cell, especially with reference to bleeding or maybe even pain? In the sense, sickle cell patients usually have the major problem as pain, but I'm wondering. Thank you.
Is Lucy here?
Yes, please.
Oh, great.
Yeah. So if I heard your question well, he said, is it having any relationship with sickle cell? Am
Lucy,
I right?
you're muted.
Akwesi, I could hear her, so she's not muted.
Yeah. Please, the question again.
Is it both questions? Is it which question do you want to hear again?
Lucy and Linda, you are both muted.
I just had one question. My volume keeps going and coming.
Akwesi, we are not muted. I am very unmuted. So you didn't hear any of the questions. You heard one. Please answer the one you heard,
yes.
And then we'll let the other person
I want to be certain. I'm not sure whether that I heard that he said, does alpha thalassemia have some association with sickle cell? Is that what he said?
Something of
the
sort.
That was what I said. And also, if there is any part of that could affect the pain, perhaps expression in patients with sickle cell, since you are talking of bleeding, which we all know is associated with some level of pain. Thank you.
Well, I did not check. I did not try to look at the association between alpha thalassemia and then sickle cell. But what I know is that alpha thalassemia patients usually have normal serum ferritin levels, even though they might show microcytic hypochromic.
Oh, Lucy's connection is not good. Lucy, can you type?
Or she can mute the video so that it will free up more bandwidth.
Okay, so as we wait for Lucy, we have one more question. That's Emmanuel. I'll give Emmanuel the opportunity to ask his question, and then we hope that Lucy comes back to answer Akwesi's question.
Thank you very much, Linda. My question goes to Hannah. I'm a bit concerned with the way the sample was collected. She indicated that she used convenient sampling. What I understand is that convenient sampling is non-probabilistic. Now, what it means is that if you want to do any analysis with a non-probabilistic sampling technique, you should confirm whether your data follows a certain distribution before she uses the probabilistic parametric analysis approach. For instance, before you use the correlation analysis, you want to ensure that it follows a particular distribution before you use the Pearson. Otherwise, other correlation analysis are recommended. I want to be sure, did she check that? That's the first one. And then the other one is looking at the way the sample was collected conveniently. Is it possible that if I consider a different sample, I'll get a different results? Is it possible that can happen? And then the last one is on the comparison with the existing studies. Is it just coincidence or that is the reality? Because I'm looking at the methods and how well your results are consistent with the existing literature. Thank you.
Thank you, Emmanuel, for the question. So, Hannah.
Okay.
Can you help?
So, Ima, thank you. So in the first place, I'm sure that because we didn't use a probability technique. We didn't use a public technique. When you use other method, you are going to get other result. Also, actually use the questionnaires were adapted from I told you it's a structured one, which was adapted and modified to suit the study objective. So per their description on that is how we're able to correlate whether this one was a good one or the consistency with it, because I actually use what they have to compare with other studies. Thank you.
Emmanuel, is that okay?
I will take it like that.
But I guess you have some suggestions. so that going forward, if they decide to recruit more
Yeah,
people,
definitely. Definitely, I should have. For instance, from all indications, it appears that everything was done at the Wejak Barry Hospital. So what it
Yeah.
means is that if I decide to randomly sample from different hospitals, it's likely the results will change. And for data collected from what she did, I think the best approach could be probably the Spearman's correlation coefficient that will better estimate the relationship between the two. The first reason is that the data that she's presenting is only frequencies. They are not measured data. And so it's reasonable to use Pearson try to do multiple regression. And that's the point. And maybe she could try to show the tests or whatever instrument was used to collect the data is consistent. And by consistent, it means that if I collect data in one area, that the results will be applicable to other areas. And this is done statistically. So she can read about how to show that a test is consistent and to improve. Thank you, Linda.
Okay.
I think this is very good information.
Yeah.
Hannah.
Well, thank you very much, Yuma. We will consider all these things before doing the publications and make into consideration. Thank you very much.
No, but if you're saying before making the publication means that you're not sampling anymore, then I guess it would be probably your limitation section. Okay, so I think I have, do we have any more? It's, okay, we still have, no, we don't have time. Actually, so I am so sorry to all those who had questions that were not read out or answered. And please, we will send email addresses of all the presenters so that you can contact them after the conference. So at this time, I would hand over for the closing and announcements to be made. Thank you very much.
Dr. Yamfo?
George, I thought you'd be talking about the yearly dues, the one that we see on screen.
All right, so if I may do a quick sales pitch at this time. So we welcome new members. There are so many benefits to these interactions, some for which we wouldn't have time to enumerate. But as we speak, we just participating in this conference, one of the presenters last year is already here in Massachusetts, starting a PhD. As we were talking, I just got a call from another younger, former participant, making progress. So there's a lot to be gained from the mentorship, from the collaboration. The dues for professionals is 50 Ghana cedis, and the dues for students for the year is 10 Ghana cedis. And we can do mobile phone payments, and the details are on there, or else we can use our EcoBank account. And all dues go to support the work that we do, whether they are workshops, training in a variety of formats, and holding events. So we'll put this up again sometime tomorrow, but just something to reflect. Thank you.
Okay, just to add more to what George said, it will interest you to know that Professor Gordon Awandari, current director of WACB, it's a member, not a former, he's still a member of COBREG. Professor Solomon Furakwa, director of WACMIC, he calls it WACMIC, WAGMC. And then we have a lot of professionals. You can see Dr. Linda Mwa, Dr. Akwa, and George Akwa Mensah, and everybody. So it's a good group to be a member, and we have a lot of mentors. So like we always say, establish collaborations so that we can all build ourselves as we go on. So overall, I think it's been a very successful meeting. The only thing I'm slightly disappointed is the number of participants, because we had over 90 people registering, but we couldn't make that number for one reason or the other. But overall, I think the quality was good and members participated fully. So that brings us to the end of today's talks, and we hope to see you again and interact with you again tomorrow from 1 o'clock Ghana time. Thank you very much.
Thank you.
Much to all the organizers. Thank you very much.
Okay, bye.
Thanks for helping me present.
Oh.
Listening.
I'm still.
Bye-bye all.